So let's look at the base calculation. In most cases, some books, they don't have this, but in most cases, we ask it. So meaning that these complementary bases. So we're going to use the principle of complementarity. That adenine goes with thymine, guanine goes with cytosine, and then we'll be able to calculate anything which is needed. Here is the first question. If adenine is 30%, what is the percentage of cytosine? So we know that adenine does not go with the cytosine. So how are we going to bring about the percentage of cytosine? So in this case, you know that percentage means out of 100. When you say that you have got 30%, it means that you have got 30 out of 100. If they say that you have got 60%, it means that you have got 60 out of 100. So now, if they say that adenine is 30%, it means that if adenine complements with thymine, if the percentage of adenine, remember James Watson and Francis Crick, they said that these are complementary base pairs. How did they find that? They found out that the percentage of adenine is equal to the percentage of thymine. Therefore, those are complementary base pairs. So if adenine is 30%, definitely thymine is also 30%. So the remaining percentage, if you add 30 plus 30, you get 100. The remaining percentage will be for cytosine and guanine. And remember, these two are complementary to each other. Therefore, if I divide by 2, then I get the individual percentage of guanine and cytosine. This is what I'm trying to say. So we are saying that adenine goes with thymine and guanine goes with cytosine. So if this is adenine, if adenine 30, then automatically, if adenine 30, then automatically thymine is also 30. So 30 plus 30, okay, I get 60. So 30 plus 30 is 60. So this 60 is for these two. So the balance from 100, because this is percentage, it's indicated here as percentage, the balance will be for this. Therefore, I will say 100 minus 60, which is equal to 40. So it means that guanine and the cytosine are equal to 40. So that when I add the 60 plus 40, I get 100, which is the percentage. Therefore, because these two are equal to 40, so I'll say that guanine and cytosine are equal to 40. So if I want to get one individual, because this is equal to this, therefore I divide by 2, then I get 20. Therefore, cytosine is 20%. That's the meaning, or that's how you can calculate it. So even if they manipulate it, you have to use this principle of the complementary base pairs, that adenine, thymine, guanine with cytosine. Let's look at another example. If cytosine is 20, what is the ratio of guanine to what is the ratio of thymine to guanine? This one is not worked out, therefore it means that I'm gonna just talk about it and then see if you can find the correct answer. If cytosine is 20%, what is the ratio of thymine to guanine? They didn't talk about cytosine here in the question. All right. So now what are you going to find? Find out the percentage of each individual base. Remember, cytosine goes with the guanine. We saw it here that guanine goes with cytosine. So if this is 20%, automatically even this one is 20%. Therefore, I'm going to use the same principle, 20 plus 20, because 20%, 20%. 20 plus 20 is 40, but this is percentage. Therefore, I'm going to subtract it from 100. So 100 minus 40 is going to be 60. Therefore, it means that cytosine 20, guanine is 20, therefore it's 20. And then what about now thymine and adenine? Because the balance is 60, therefore it means that adenine and the thymine is equal to 60. If I divide by 2, I get 30. So this is 30, 30, 20, 20. So that if I add, I go back to 100. 20 plus 20 plus 30 plus 30, it gives me 100. So if you add and you don't get the actual total number, then it means that your answer is what? Is wrong. So now, what am I going to do? I'm going to say that if this is 30 and this is 20, because we saw that guanine goes with the cytosine, which is 20 here. Therefore, this is 20 and this is 30. So if I divide it by 10 to get the simplest ratio, I'm gonna get 3 to 1. So your answer must be 3 to 1. And this must be corresponding to this arrangement. Don't say that guanine to thymine, then your answer will be wrong because you have set your own question. The question is the direction is saying that thymine to, or the arrangement thymine to guanine, not guanine to thymine. So we can find this question in objective or in when you are just writing a small subsection. So please don't forget to know how to calculate this. Then another question, you can also practice it. If DNA has 5,000 bases, only 500 is guanine. What is the percentage of thymine to cytosine? Drop your answer in the description below, and then I'll come back to you when I see that there is big challenge in that. So if not, try to contact that number here so that you can be helped just in case that you don't get it. Or you can try to find your teacher and then you'll be able to help you out. Let's look at DNA replication. What is DNA replication? Soon we'll be having an online discussion with the whole country. I'll be letting you know in the dates where we're going to have the online discussion. You ask any question you would like to know, and then I'll be able to answer those questions. Even if you're asking the question ahead, but I'll be specific. I'll give you specific topics that, okay, you can look for questions from this topic to this topic. I'll be able to answer those topics, those questions in real life. Thank you very much.
This is a channel which is going to help you to obtain a distinction in life science. Keep in touch. We will be posting each and every section of life science until the end of the syllabus. Thank you. Let's look at DNA code of life. When we talk about DNA, we know that we have been using this in the movies. You have been seeing this in the movies about DNA. But what is DNA? If you remember in grade 10, we covered some nucleic acids whereby we talked about DNA and RNA. So in this case, we are going to look at it in detail so that we know exactly what is DNA. This is one of the topics which you can answer very well and get all the marks. It is found in paper 2. So it means that you have started your journey, you have started it with paper 2. Make sure that you understand this topic because one topic links to another topic. If you don't understand one topic, then it means that next topic will be a challenge for you to understand. Let's start without wasting time. So you'll be able to find us on YouTube, on Facebook, and our website. So you'll find the material and you'll find videos which we'll be posting. Don't forget to subscribe so that you don't miss out for this time. Let's look at DNA. Introduction about DNA. DNA is often referred to as a code of life. Why? Because it contains instructions on how to build various proteins. You know that our body is made up of proteins. Therefore, if our body is made up of proteins and what makes protein to be made is DNA, we say that DNA codes for life. So let's look at where is DNA found. If you look at this, you'll find out that DNA is found in the nucleus. This is the structure of the nucleus, if you remember in grade 10. So basically here we are looking for the nucleus. So the DNA we can ask you a question that where is DNA found? So basically here we can see that DNA is found in the nucleus. So if you look at it, we call it chromatin network. Remember, when the chromatin network condenses, it forms what called the chromosomes. If you uncoil and go into detail of the chromatin network, you'll find what called DNA. But what is DNA in full? Because you can ask yourself, you have been talking about DNA. What is it in full? We're going to see it in detail. What is DNA in full? So we say that if you magnify, you'll find out that if these chromatin network condense, they form what called chromosomes. If you look at the chromosomes, if you make it bigger, or if you try to magnify it, you find the DNA is found there. But we shall see that DNA is made up of different bases and different nucleotides. We are going to look at them in detail. Don't stress too much. You will understand it better as we continue. We have seen that DNA is found in the nucleus. So when I talk about chromatin network, I talk about chromosomes, I talk about DNA, I talk about genes. Those are the same thing, but on different scale. For example, I can say, where do you stay? You can say that I stay in Africa. Where exactly in Africa? In South Africa. Where exactly in South Africa? In Gauteng. Where exactly in Gauteng? In Johannesburg. Where exactly in Johannesburg? In Florida. Where exactly in Florida? So you talk about different scales. So when you talk about chromatin network, talk about chromosome, talk about DNA, talk about genes, almost the same thing, but at on different scale. However, when you are defining, you can define them differently. So let's look at the nucleic acids, because we're seeing that this DNA is an example of a nucleic acid. So let's look at the nucleic acids. Nucleic acids, it means that the acids which are found in the nucleus. So we are saying that these nucleic acids, we're saying that consist of nucleotides. These acids, they have what called nucleotides. They are building blocks. They are building blocks, or the monomers. So sometimes they can call you, they can say the smallest unit of a nucleic acid is called what? It's a nucleotide. The smallest unit of DNA is called what? It's a nucleotide. Those are questions we can ask you in an exam. The smallest units, or what are the monomers of DNA? What are the monomers of nucleic acids? Those are the nucleotides. Don't forget that, the meaning of the nucleotide. So let's look at it. A nucleotide is made up of, number one, a phosphate group. You have to know this. A phosphate group, yes, the phosphate group is represented as a circle, and then is made up of a sugar. Basically, the sugar it depends on which type of nucleic acid. It is either deoxyribose or it's a ribose. When we talk about deoxyribose, it means that D means negative. Oxy means oxygen, ribose. It means that this kind of sugar lacks an oxygen somewhere. That's why you call deoxyribose. So, it's made up of ribose, a sugar which you call ribose. It means that this one is full. I don't need to explain that much. So it means that it is just full. Yes, you will study these sugars more when you go to university. So because this sugar has 5 sides, therefore this kind of sugar is called a pentose. Penta means 5. So it's made up of 5 sides, which means penta. Yes, so because it is a sugar, OSE indicates a sugar, like saying glucose, yes, maltose, ribose. So because this is a sugar, so it is the type of sugar, yes, is called a pentose. But what is its name? Because of 5 sides, therefore it's called a pentose. But what is its name? It is either deoxyribose or it is ribose. So in exam, what are we going to ask you? We are going to ask you, name this sugar. Don't call it a pentose. If it's DNA, tell us it is deoxyribose. If it is RNA, tell us it is ribose. So we shall see when you go to the questions very well, so that you can answer these questions without stress. Another one is called nitrogenous base. So nitrogenous base, there are 5 types. We have what called the adenine, the thymine, the guanine, the cytosine, and the uracil. If it is DNA, then it means that it's going to have some specific nitrogenous bases. If it is RNA, it's going to have some specific nitrogenous bases. So we will see which one goes to the DNA and then which one goes to RNA. So these nitrogenous bases, they look like this, or sometimes you can make it a different shape, depends on which type of nitrogenous base is that. So basically, we see that the structure of nucleic acid is made up of a phosphate group, is made up of a sugar, and made up of a nitrogenous base. Which kind of sugar? It will depend on which type of nucleic acid is that, either DNA or RNA, and which kind of nitrogenous base is it having. It will also depend on which type of nucleic acid is it. Is it DNA or is RNA? So sometimes they can ask you, draw the structure of the nucleotide. So the structure of the nucleotide looks like this: a phosphate, which is represented with a P, yes, a circle, or it's a P, and then it is bonding with the sugar, which is the S. Yes, this is a phosphate sugar bond or sugar phosphate bond, and then the sugar is bonded to a nitrogenous base. So this is the structure of a nucleotide. In exam, you don't need to shade this. How do you draw it? You just draw it like this: P, S, and N, depending on which type of nitrogenous base, so that it will take the shape. However, even if you draw it as a rectangle, it still is fine. But this one must have five sides because it is a pentose, and this one must be a circle. So that's how you can draw it in exam. So it means that now you have marks already. You have questions you can answer. If you visit the 2020 November December exam, you'll find out that they brought this question, whereby you need to describe this, and you'll be able to get marks here. Yes, each point you speak is a tick. Then let's look at the types of nucleic acid. As we say that there are two types of nucleic acids. Number one, DNA, that is deoxyribonucleic acid. We call it deoxyribonucleic acid. What is it? Deoxyribonucleic acid. This acid found in the nucleus lacks oxygen somewhere. This acid found in the nucleus has a sugar, which is a ribo, and this sugar lacks oxygen on a certain carbon atom or somewhere. So you basically, no one is going to ask you to explain all this, but I am trying to explain to you so that you can understand what is DNA in full. Yes, so that is deoxyribonucleic acid. When you are writing what is DNA in full, you don't add SE here. No, you don't add SE because this is not a sugar. It is a molecule which contains that sugar. That's why we don't say deoxyribose. When you say deoxyribose nucleic acid, you get it wrong. It is deoxyribo. There is no SE here. Deoxyribonucleic acid. So these acids found in the nucleus, they have a sugar which lacks oxygen somewhere. That's the meaning. Then we have another type which called RNA, which is ribonucleic acid. This molecule has it's just a complete molecule. So it means that when you are writing, don't say that ribose nucleic acid, because this is not a sugar. It's a molecule which contains a sugar. So we say that it's a ribonucleic acid. You remove that SE. If you say ribose nucleic acid, your answer is going to be wrong. So you're supposed to be ribonucleic acid. So we have two types of nucleic acids. Number one, you have DNA, and then number two, you have RNA. Those are the two types of nucleic acid. So we're going to look at this in detail. And this DNA is the master of all. So it controls all the activities which are happening in your body. All structures which are developing in your body are being controlled by DNA. So that's why you say that DNA codes for life. Why? Because it controls all activities. Without DNA, you are nothing. If there's no DNA, our cells are useless. So with the DNA, then we find ourselves there. And if any problem happens on DNA, it's going to affect your physical appearance. We will see this as we continue. DNA. Let's look at it. Where is it located? The types of DNA we have depend or will be determined by where these DNA are found. Number one, we have nuclear DNA. It means that it's found in the nucleus. It's called the nuclear because it's found in the nucleus. You can't call it nucleus DNA. No, when you're adding something on the nucleus, then it becomes nuclear. It must end with R. Yes. Then you have mitochondrial DNA. You don't call it mitochondria. If you're adding something on the mitochondria, it becomes mitochondrial DNA. This one is found in the mitochondria. And then you have choroplastic DNA. This is found in the chloroplast. They brought this in 2020. They brought this in 2020 exam, national exam, and people couldn't remember this. Use this material is going to help you to write correct answers, and you'll be able to write without stress. All the information in these slides is very important. Watch the video. If you don't understand a certain concept, watch these videos so that you can understand. Even if the teacher is not there or is having a problem, still you can use this material and you'll be able to pass your exam. That's why we are designing this material for you. So we have nuclear DNA found in the nucleus. You have mitochondrial DNA, which is found in the mitochondria, and then you have choroplastic DNA from the chloroplast. So if you look at it, it means that the type of DNA will depend on where they are found. So nuclear DNA and the mitochondrial DNA, they are found in both plants and animals. While choroplastic, because chloroplast, it makes the plant to look green because of its component, which is chlorophyll, then it means that choroplastic DNA is only found in plants or other green organisms. Yes. So they can ask you, name two where you can find DNA in animal cell or in animals. Therefore, the answer is going to be nucleus, and then another answer is going to be mitochondria. Don't confuse the type and the area where that type is found. Students always confuse this. Please don't confuse where DNA is found and where and the type of DNA. All right, let's continue. How is DNA discovered? How did you come up with this DNA? History of discovery of DNA molecule. So here we're going to just look at just the basics, how DNA is discovered, just the small basics of how DNA is discovered. All right.
Welcome back to our class. We are going to look at the different people who were involved, the major people who were involved in the discovery of DNA. James Watson and Francis Crick, these two, they were working together to try to solve the problem of this DNA molecular structure. As they were working, also other two people were also working on the same structure, that is Maurice Wilkin and Rosalind Franklin. Before I continue, let me just be clear. James Watson is a guy, Francis Crick is a guy, Maurice Wilkin is a guy, Rosalind Franklin is a lady. By the time this DNA was being discovered, ladies were not regarded as people who can do something. So in actual sense, there were some issues of gender. So Rosalind Franklin was taken to Maurice Wilkin just to be an assistant. So Rosalind Franklin worked with Maurice Wilkin, and Rosalind Franklin discovered the structure of DNA using X-ray diffraction pictures. So without the concern or without the knowledge of Rosalind Franklin, Maurice Wilkin took the picture to James Watson. In actual sense, Maurice Wilkin used the Rosalind Franklin pictures and then took them to James Watson. Once James Watson and Francis Crick, the moment they saw the picture, then they had to figure out, or it awakened their brain, and they got an imagination how the DNA structure looked like. So they use that information to find out the further information about the structure of DNA. Unfortunately, Rosalind Franklin died before a Nobel Prize was given. But before she died, she published an article about DNA structure. The same time James Watson published the article of DNA structure. Before she died, she published an article concerning about DNA structure. Also, James Watson also did the same. But because by then people were not recognizing ladies too much, so they took James Watson's paper to be on top, and then Rosalind Franklin paper to be on bottom. And then what happened? They recognized James Watson, that is the one who published first. Therefore, the credit goes to James Watson and Francis Crick, other than Rosalind Franklin. Yet it was the hard work of Rosalind Franklin to be the person who started with the discovery of this, because James Watson used the X-ray refraction pictures to explain more detail concerning about this. So this shows that we should not undermine ladies, that they cannot do anything. They have ability to do the work guys can do in terms of education. So we should stop gender issues. We thought that ladies cannot do anything, which is not right. Even up to now, this perception is still available that ladies cannot do anything, but this is wrong. Why? We have a lot of things which are being made by ladies, and these ladies, they are helping in our community. And so this is just a clear indication that that even ladies can do something which is important in science. So we want to see more scientists who are ladies coming out like Rosalind Franklin. So unfortunately, Rosalind Franklin died at the age of 32 before the Nobel Prize was given. So in 1962, that is James Watson, Francis Crick, and then Maurice Wilkin, they received a Nobel Prize determining the molecular structure of DNA, how DNA is a double helix with complementary base pairs. But if Rosalind Franklin was there, then it means that Rosalind Franklin would have obtained a Nobel Prize instead of Maurice Wilkin. Why? Because Rosalind Franklin did a great job than than Maurice Wilkin, because Maurice Wilkin just stored the information of Rosalind Franklin and took it to James Watson and Francis Crick. So how do we? Let me just repeat. James Watson and Francis Crick, they were working together to find out the molecular structure of DNA. Rosalind Franklin and the Maurice Wilkins, they're also working together. They found out the structure of DNA using the X-ray diffraction picture. This structure was taken by Maurice Wilkins to James Watson and Francis Crick, and then was used to discover by James Watson and Francis Crick to find more that DNA is a double helix with complementary base pair. They found out that the DNA consists of adenine, thymine, guanine, and cytosine. And they found out that adenine, the percentage of adenine is equal to the percentage of thymine, and the percentage of guanine is equal to the percentage of cytosine. Therefore, they say that adenine complements with thymine and guanine complements with cytosine. Therefore, they say that these are complementary base pair. This is how this came about. This that it is a complementary base pair. This forms, because there are two strands, it forms is a double, and then these strands they twist around because it is in a wave form. Yes, it forms a helical. Because it is two strands or two helicals, then it forms what we call a double helix. And then say that DNA is double helix with complementary base pair. That's how they came about with the structure of DNA. How do we ask in exam? How do we ask in exam? Who discovered the structure of DNA? That is James Watson and Francis Crick. Who discovered the structure of DNA using the X-ray diffraction pictures? Then that is Maurice Wilkin and Rosalind Franklin. In which year was DNA discovered? That is 1962. Which one of the following obtained the Nobel Prize of the discovery of DNA? Crick. Then they can give you different options, but in this case, they can. You have to know these two names because we can mix. James Franklin and then Maurice Watson. In this case, you have to know. Yes, because you see Maurice, you think that that's the person. Yet they say that it is Maurice Watson, whereby that person doesn't exist in this case. So James Watson, Francis Crick, Rosalind Franklin, and then Maurice Wilkins. Yes, it's supposed to be like that. You have to know them. Don't confuse them. Some people say that these names are complex. How many people do you know in your class? There are so many people you know, but you know them, their names. If you're a soccer supporter, how many people do you know? You know almost the whole team, but these are just the 4 people whereby you can still memorize these people, and then you are able to get marks just in case that a question has come. So that's how we can ask questions concerning about this. After this, we will look at the questions, more questions which are regarding to this topic. Then let's look at the detailed structure of DNA. DNA profiling. What is DNA profiling? DNA profiling, when you are going for DNA testing, you are going for DNA test, we are just going to do what you call DNA profiling. So we make the profiles of DNA. So how is it identified? You use the, it is shown on the screen as bars. So if the bars are corresponding, then it means that those organisms or those individuals are related to one another. So DNA profiling is a technique used to identify someone's unknown DNA profile using a known DNA profile. For example, you are looking for your father, and then you don't know your father. There are 2 guys who have come, and these 2 guys, they are claiming that you are the son or you are the daughter of those guys, but only one guy will be the father of that daughter or that kid. So now I know the DNA profile of you, I test the DNA profile. So your DNA profile becomes the reference point. So that is the known DNA profile. So this known DNA profile, I compare it with these 2 people whom I don't know their DNA profile. I go and then I test their DNA. If I find that one of them corresponds with my DNA profile, then it means that that is my father. If the 2 of them, no one corresponds to my DNA profile, then it means that nobody there is the father. So that's how this technique is used. Is a technique we use to identify someone's unknown DNA profile using a known DNA profile. So you are the known DNA profile, the father, the other 2 guys are the unknown DNA profile. So if they correspond, then we say that you are the father of that kid. So here is an example. Yes, for example, this is a scene, crime scene, someone committed a crime. Therefore, when I go to the crime, I find some material, for example, hair, blood, I collect that sample. When I collect that sample, I take for DNA testing. or for DNA profiling. So I get the profile of the DNA. That's why in most cases, when there is a crime scene, we don't allow people to go there. Why? Because you might bypass there, and then when they come to collect the sample, your hair is included there. And then you're going to be one of the suspects, because your DNA, once they test there, they see that you are among the people who were there. That's why if there is a crime scene, don't tend to go there if you are not an investigative person. All right. So you're saying that the crime scene, this is the sample they picked from the crime scene. So here we have the suspect. We have 3 people whom we are claiming that these are the people who commit the what? The crime. So what they do is to get their DNA profiles, and then I start to correspond with what we found on the crime scene. So if I use a ruler and then I draw here, there is nothing here. I draw this corresponds, that's 1. This one corresponds, that's 2. There is nothing here. There's nothing. Then this one is 3. This one, there's nothing corresponding. And then this one is 4. So this one is gone. Then what about this? This one is corresponding. 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 This one is also corresponding. So all of them are corresponding. What about this? Shows that there is some which are corresponding and some are not corresponding. Therefore, we will conclude that the suspect number 2 is the person who did the crime. Why? Because what we collected from the crime scene and his or her DNA profile are exactly the same. Therefore, suspect 2 will be taken for accountability of this. So what are some of the uses of DNA profiling? What are some of the uses of this technique? What are some? Yes. Okay. Number 1, it is personal identification. You know about the person someone is missing. So it's very easy to identify the person, the identity. Someone has burned somewhere, so it's easy to identify the identity of that person. Then number 2, paternity and maternity. Maybe your mother, I don't know your mother, and there are 2 ladies who are claiming that you are the mother. They are the mother, so or father. We can use this technique to identify the actual parents of this. How is this one possible for mother? Maybe in the hospital, and they have swapped the kids. You know, when the kids are just born, in most cases they don't tend to resemble their parents, but as they grow on, they turn to resemble their parents. So by that time, still we can do what? We can find the parents there. Then another one is diagnosis of inherited diseases. Diagnosis of the inherited disease. There are some diseases which can be diagnosed using DNA replication to know actually that this is the disease you have using DNA. Using DNA profiling. So diagnosis of inherited disease, and sometimes we can even find a cure for those diseases. Then another one is criminal identification. Criminal identification, for example, this on the crime scene, they go take the samples which are there, and then they compare to the person they are suspecting, and then we find the person who did the crime. Then forensics, maybe someone is burned still, maybe in an accident, you can't find anything, so we can still use DNA even if it is 20 years. As long as I get any remain of you, still we can find the DNA from there, and then we can correspond it to. Then they are saying that identifying suitable organ donor. It's not easy to find the organ donor. So you have to first see that if the DNA profile of you and the person you are giving the organ is compatible or is almost the same, or otherwise the organ will be rejected by that person's body. So what we do is to first identify to see the correlation between you and the person who is going to receive the organ. And then once they are compatible, then they transfer the organ from one person to another. Then what are some of the disadvantages of this technique? Number 1, It is expensive. Not everybody can achieve or can afford to do this. Not everybody. So it is very expensive. So it requires people who are, yes, well off. Then number two, human errors. Some people imagine someone is drunk, some maybe a bear is drunk and then go back and then is trying to analyze. Definitely is going to have some errors. Even if it is not that, still when you're analyzing, you can have some, you are not 100% correct. There are some errors which are developed there. Yes, that's why we increase the sample size, and then also we repeat the experiment so that you increase the reliability, so that you increase the sample size. So it means that you have to be with a lot, and then you have to repeat this technique so that you are sure that what you got is exactly even what another person is gonna get. Then we shall say that that's maybe the human errors have been minimized. Not all hospitals have necessary requirement to carry out this DNA profiling, just like schools. But look at the schools, you'll find out that some schools, they have better equipments, they have almost everything students need to learn, but other schools, they are struggling. Whether they are government schools or where they are private schools, you will find them that they have different categories or different degrees. What about now hospitals? Still, some hospitals are well off, they can have these techniques to be supported, others they don't. Others, they have equipments, but they don't have the chemicals they use for this to happen. So you see that some hospitals, they don't have necessary requirements to carry out this DNA profiling. So then it is possible to plant DNA at a crime scene, giving false evidence. Yes, it is possible. If someone wants to frame you, then automatically can be planted, and then it means that this is not a good idea of DNA profiling. So, innocent people or persons' DNA might be at this crime scene, at the scene or crime scene, even though they had nothing to do with the crime. That's why I say that you are just passing, and then your hair drops, and then when they're collecting the sample, then they also take your hair. Then you are innocent, but because the DNA profile is showing that it's you who was there by the time the crime was committed, then automatically you're gonna be among the people who are suspects. So and that's why we are saying that, yeah, DNA profile has some disadvantages. So let's look at RNA, ribonucleic acid. So once we talk about RNA, then it means that we are going to start what you call protein synthesis. So RNA and DNA are almost the same, but they differ in some few aspects. Why am I saying they're the same? Because both of them, they are nucleic acids. There's 3 types of RNA based on the function they do. So there are 3 types of DNA based on the location. But RNA, there are 3, but these ones, they are based on the function they are doing. So number 1, we have messenger RNA. As you hear the word, it is a messenger responsible for carrying genetic information that is transcribed from DNA. So it carries it from DNA and then takes it to cytoplasm or to the ribosome so that protein synthesis can be made. Then number 2, you have ribosomal RNA. That is, it means that it is forming the ribosome. It forms the skeletons of the ribosome. So this one forms the ribosome and produce the proteins based on the information received from the transfer RNA. So the transfer RNA, when it's coming, it comes to the ribosome. So the ribosomal RNA is just the skeleton of ribosome. Then you have the transfer RNA. This one changes, or it has what called the anticodons. It code for the specific amino acid. So it transfer the amino acid from the cytoplasm and then brings it to the ribosome so that a protein is what is formed. We will see this when you go into detail of protein synthesis. So what is the role played by RNA in protein synthesis? Number 1, messenger RNA molecules carry the coding sequences for protein synthesis. Number 2, ribosomal RNA forms the core of the cell's ribosome. And then number 3, transfer RNA carry amino acids to the ribosome. That's what I've been trying to explain, that their function determine the type of RNA it is. What they do will determine what that type of RNA is. So there are 3: messenger, ribosomal, and then transfer RNA. So what is the structure of RNA? Structure of RNA is almost like DNA, but just a small difference of uracil instead of thymine, and then a single strand instead of double strand. For it is straight, it's not helical. So saying that a single-stranded molecule consists of nucleotides. We saw that it's also containing nucleotide, phosphate, sugar, and then nitrogenous base. But the phosphate is fine. Sugar is ribose instead of deoxyribose, and nitrogenous bases, it has uracil instead of thymine. Nucleotide is made up of sugar, that is the sugar. Which type of sugar is ribose, and then phosphate and nitrogenous bases. It has a phosphate nitrogenous bases, and then 4 nitrogenous bases of RNA are adenine, yes, adenine, uracil, cytosine, and guanine. So these are the 4 nitrogenous bases regarding to RNA. So this is the basic structure of RNA. So this is the messenger RNA. This is how messenger RNA looks like, and then this is the transfer RNA, how it looks like. Then the ribosome RNA, it just forms the ribosome, and basically for this level you don't need to know its structure. Yes. Let's look at protein synthesis in detail. Let's look at protein synthesis in detail. Thank you very much for coming back to our channel. Today we are going to be looking at DNA replication. DNA replication. So what is DNA replication? How does it occur? Why does it occur? Where does it occur? So that's some of the questions we ask on DNA replication. What is DNA replication? To replicate, it means that it is doubling, to double. So like making a copy. So that is the meaning of replicate. But the old and the new must be exactly the same to bring about the meaning of replication. So what is DNA replication? Is the process by which DNA makes exact copy of itself. So when you are writing a definition and you don't talk about DNA making exact copy, if the word exact is not there, then it means that you won't be able to get a tick. So it must have the meaning of, it must have the word exact. So that's why you say that the old DNA molecule must be exactly the same as the new DNA molecule. Then you're saying that when, now the question is when does it occur? When does, when in the cell cycle it takes place? When does it occur basically? Or it occurs during interphase. Yes, you can say during interphase or before cell division, because interphase occurs before the cell divides. Therefore, it is here you have generalized. If you say that cell division, you have generalized. Then interphase, you are just specific to when exactly does it occur, because we have different phases, but the first phase is interphase. So DNA replication occurs in interphase. So where, now another question is about where does it take place, or where does it occur? Then you are saying that it occurs in the nucleus. So it means that DNA replication occurs in the nucleus. When you're talking about DNA replication in this case, we're talking about DNA replication in eukaryotic cells. We're not talking about DNA replication in eukaryotic cells or in prokaryotic cells because these don't have nucleus. Then why does it occur? Why should we have DNA replication? Sometimes we ask the significance of DNA replication. What is the significance of DNA replication? That is the same as asking you, why does it occur, or why does it take place? It takes place to double. You want to see the word double? Yes, to double. What is it doubling? The chromosome number. Don't say to increase the chromosome number. Yes, it's correct. But when you're mentioning, you want to see the word doubling. To double the chromosome number or to double the genetic material so that now each cell can obtain an exact number of chromosomes. So why does it occur? The answer we need is to double the chromosome number. So if you had 23, if you were haploid, then when you double, then you become diploid, so that now the cell divides. If it is case of meiosis, divides, then goes back to haploid, so that you maintain that chromosome number from generation to generation. Yes. So another one is results in the information of identical daughter cells during mitosis. Remember, cells undergoing mitosis are diploid. If they are diploid, it's supposed to double. The genetic material is supposed to double. When doubles, the cell must divide. Once it divides, then it means that each cell must have the same number of chromosomes. So it will be identical to the original cells. So it means that if I have 23 pairs in one cell, then it doubles, it's going to become 46 pairs. So when it divides by mitosis, it's going to go back to 23. So that is the meaning of to bring about to identical daughter cells during mitosis. So in meiosis, it is always haploid. Then when it duplicates, then it becomes diploid, whereby it is unreplicated. Yes, it is single-stranded. Sometimes call it unreplicated. But after replication occurs, then it becomes double-stranded chromosomes. So it becomes double-stranded chromosomes. We will see this when we go to meiosis. So in exam, when they ask you what is the significance of DNA replication, they brought this question in 2020 November, December still, but in most cases, students, when you ask such questions, they always find a challenge to answer this question. Then how does it occur? In other words, the process of DNA replication. How does it occur? How does DNA replication occur, or the process of DNA replication? So you have to first be with an imagination in the head so that you can describe the process of DNA replication. You need to first know this, the imagination of the structure of DNA. Then you have to follow the diagrammatic representations, and then you form the two new DNA which are genetically identical. So using that diagrammatic representation, from there, then you can write the points which are needed. All right. DNA is, we say that is a double helix. Yes, a double helix. It means that is two helical, first helical, and then the second helical. So it has two strands. So since we want to carry out DNA replication, this helical must straighten. It must form two strands. Yes. So it must try to break. When it uncoils, it forms this. But I'll show you how do we write in exam. So it forms two strands. Yes, two strands. After forming two strands, now these bonds are supposed to break. So you can say it unzip, or you can say weak hydrogen bonds break. So you can use any of the two because they mean the same. So that is unwinding. It starts to form these two. So now it means that you have two strands which are open. Yes, it was like this. Then it, because it was coiled like this, so it uncoils, so it forms these two strands. When these two strands, if you bring it here, yes, it opens. When the weak hydrogen bonds between these bases break, then it exposes these two strands. Then, if these two strands, when they uncoil like this, then the free nucleotides of DNA from the nucleoplasm will come and arrange. When they arrange, they are forming now another strand on it. This strand must be complementary to this strand. So I have the new strand and I have original strand. Yes, so it means that what is here on this DNA which is forming must be exactly to what is this DNA which is also formed, must be exactly the same to this previous DNA. So basically I have three kinds of DNA here, but one is splitting to form two. So this is the which is original, so it means that it has this original strand. Still, this is original, it has this original strand, and then it has this new strand. So at the end, I'll have two new DNA. So I have two new DNA. So it's gonna form like this, whereby it has the original, the original strand here. It has the original strand here, and then it has the new strand. So then it means that you form two new DNA. How do you write in exam when you're asked to describe the process of DNA replication? DNA double helix. So you start by writing the DNA double helix. Yes, it unwinds the weak hydrogen bond. When it unwinds, it forms two strands. So now the weak hydrogen bond break. So I say that you can say weak hydrogen bond break, or you can say unzip. So when you mention one of that, you get a tick. To form now two strands, it forms two strands. Yes, because I want to form two new DNA, then each strand will act as a template for formation of a new strand. So template is like a mirror. Yes, so the old strand is acting as the object, and then the mirror you form another one which is inverse you are seeing. So now this old strand will act as a template for formation of a new strand. Even this one will act as a template formation of the new strand. What do they use to form the new strands? They use the free floating nucleotides of DNA from the nucleoplasm. So it forms two new DNA which are genetically identical, each with one original strand and then one new strand. And then this process is controlled by enzyme. So that's basically what you are supposed to explain when you are in exam. So let's look at it. When we are writing, each different color means a marking point. So you should not forget about it. DNA double helix, we give you a tick, unwind. Yes. Weak hydrogen bonds, they break. Yes. Weak hydrogen bond break, we give you a tick. Or DNA double helix, unwind, and then unzips. Then you also give a tick here and give a tick here. So any of that, you will get a tick. From to form two separate strands, you will need to see the word two, and these two strands must be separate. So you say to form two separate strands, or to form two single strands. So you get a tick for that, and then you're saying that each acting as a template. What is each? Each strand you have mentioned here is acting as a template. If you don't say each, you say two strands. The two strands formed are acting as a template. We want to see that word, either each or the two strands. We must see that. If you don't mention that, then you lose a mark for the template. Using, what do they use? Using free, want to see the word free, floating nucleotides, free floating nucleotides from the nucleoplasm. So when they use free floating nucleotides from the nucleoplasm, what happens? They form what called a complementary DNA strand is formed. As I showed you that it has the new strand. So the new strand becomes the complementary strand to the original one. So it forms the complementary strand, whereby adenine pairs with thymine, and then guanine pairs with cytosine. the two new DNA molecules are formed, whereby these two new DNA are genetically identical. So it forms two new DNA, and each DNA is identical to the other. So basically, the two new DNA are exactly the same. That's why I said that DNA replication, we want to see the word to form exact copy, exact. So that if they are genetically identical in terms of genes, then it means that it won't affect the physical appearance of an individual. So the person won't keep on changing and changing and changing. So it will remain the same. That's why it is not affecting the physical appearance. Then you're saying that with one original strand and one new strand. So the DNA, the two genetically identical DNA formed, each is having one original strand, and then each is having one new strand, as I explained before. This process is controlled by enzyme. So basically, that's how you are supposed to write. Here we give you a tick, 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 give you a tick, a tick, a tick, a tick, a tick, and then a tick. Most sometimes we also give you a tick here, but sometimes we don't give, depending on the marking guideline. So that is basically the process of DNA replication, and that's how we are going to ask you. What about DNA profiling? Let's look at the detailed structure of DNA. In 2020, November, December exam, Life Science Paper 2, they brought this question. It was a 20-mark question. So describe the structure of DNA. This was out of 8 marks. So we say that we know that DNA is made up of nucleotides. Remember the structure of nucleotide? So it's made up of nucleotides. Nucleotides are the monomers, the building blocks of DNA. We see that all individuals have the same type of DNA, but this DNA is different in terms of what they code. That's why we have different characteristics. How is this possible? Yet all the DNA is made up of the same kind of nucleotide. Here is the question. How do buildings differ, yet all of them, they have the same material which build them? Yes, remember DNA is made up of nucleotides. So the arrangement of these nucleotides will bring up the different structure or the different composition of this DNA, or the different genes they determine. So it's the same thing. If all buildings are made up of the same type, the same kind of material, maybe bricks, but the way how you set these bricks differ in these different buildings, and then they make them to be different. But if you break them down, you find out that they are made up of concrete or cement, or mixed with water, mixed with sandy bricks, iron bars. All those things, you find them in all buildings, but the way how you arrange them make them to be different in shapes. So that's what happens in the DNA. The arrangement of these nucleotides make the DNA to be different from each other. That's why we see that all of us, we are different from each other. Even identical twins, they are different. How? You find one is tall, one is small, one is big, one is more clever, one so like that. So they are different. Even the so-called identical on my side, I don't call them identical because they are not exactly identical. They're just halfway. They are partially identical. Okay, let's continue. So each DNA is made up of nucleotide, is a tick, and then each nucleotide is now tell us what is the nucleotide made up of. Each nucleotide is made up of a phosphate group, a deoxyribose sugar, because it is DNA. If it was RNA, then talk about ribose sugar, deoxyribose sugar. Be specific. Don't tell us that it's made up of a pentose. Is made up of a sugar. Which kind of sugar? Which kind of pentose? Made up of deoxyribose sugar and nitrogenous bases. Nitrogenous bases, yes. and the nucleotides are joined together by phosphate sugar bond to form a strand. So we have seen that it's made up of nitrogenous bases. So there are four different categories of nitrogenous bases. Mention those nitrogenous bases. There are adenine, thymine, cytosine, and guanine. So if it is RNA, then you talk about adenine, uracil, cytosine, and guanine. So these bases, they are complementary to each other. So in the case of DNA, they are complementary to each other. We saw it when we are talking about the discovery of DNA, whereby adenine goes with thymine and then cytosine goes with guanine. Doesn't matter when you say thymine goes with adenine or guanine goes with the cytosine. It's the same thing as long as you compare adenine with thymine and then cytosine with guanine. Then still you get a tick. The nitrogenous bases are linked together by weak hydrogen bonds to form two strands. So we saw that, yes, these are linked together to form two strands. So if it is a double helix, it means that with two strands. So the two strands, they coil or they twist to form a double helix. All right. They're saying that the two strands, they twist to form a double helix. So basically, that is how you are supposed to describe the structure of DNA, and then you get a mark. So here we give you a mark, here we give you a mark, here we give you a mark. A mark, each different color, you give you a mark because this one has been mentioned here. Therefore, you ignore that. This is a mark. Yeah, identifying them, they are complementary. Also give you a tick there, and then we give you a mark here, weak hydrogen bonds to form two strands. It means that DNA is double-stranded. Yes, is a mark, and then this double helix is also a mark. That's how we can mark you in exam. Make sure that you don't skip these points which are in different color, because those are the marking points we are looking for when we are marking. Sometimes people, they say that they have killed the paper. If the paper has killed them, they come out of the paper and say they have written a lot, yet there are no points. Some people, they write a lot, many pages, and after that they come back with the cartoon marks. They say, why am I getting these small marks? It's not about how much you write, but it's about the quality you have written, meaning that the points you have given are the points we are looking for, the facts. We are looking at the facts we need. There are some facts we are looking for when we are marking. So if you skip those facts, even if you write a cloud of words, still you will get 0 from that. So note out when you are using our resources that these marks we are looking, these are the marks. The colored part is a mark we are looking for. If you don't have these notes, request them from that number below, then you'll be able to get the points. This is the structure we are talking about, the structure of DNA or the molecule of DNA. DNA, we said that it is a double-stranded molecule. Yes, it has a nucleotide. If you look at this point, is a nucleotide. It's a nucleotide. Each nucleotide is made up of a phosphate, sugar, and nitrogenous base. Yes, these nucleotides are joined together by a sugar phosphate bond to form a strand. Yes, the two strands, you have 4 bases: adenine, thymine, guanine, and cytosine. Adenine goes with thymine, guanine with the cytosine. The two strands are linked together by the weak hydrogen bond. So the double helix, we have not shown it here. So these are the points which you need to know. Sometimes we just bring a structure, we hide this, then ask you what is this, what is this. Sometimes we hide this and ask you what is it, because this side I have adenine, then this side I will have thymine. Sometimes we don't have anything here, but we bring this and then we put it here. So we ask you, what is this? So automatically this shape will indicate adenine. The shape which shows like this indicates the adenine. Therefore, if this shape is adenine, definitely this will be adenine, and then obviously this side will be thymine. So basically, that's how you can try to identify the structure. We will see more questions when we wrap up with this topic, so that you can know exactly how do we answer those questions, and you'll be able to get marks when you go to the paper. There is no way how you can fail if you are using these resources, because whatever we are talking about is what we said. So DNA, we said that is a double-stranded molecule with a weak hydrogen bond. Yes, and then we said that maybe characteristics of DNA still You can write them like that. It has what called a deoxyribose sugar. Yes, it has nitrogenous bases. You tell us the nitrogenous bases. The nitrogenous bases are complementary to each other. Adenine goes with thymine, guanine with cytosine, and then in the actual sense, DNA is double helix. So it's double-stranded, and then this double-stranded, it forms what called a double helix. So if they bring questions concerning about the difference between DNA and RNA, so it means that you can use this point. It's double-stranded, then the other side automatically it's not double-stranded, it's single-stranded. And then this one is weak hydrogen bonds, because single-stranded has no weak hydrogen bonds. This one has weak hydrogen bonds, no weak hydrogen bonds. If this is deoxyribose sugar, then the other side is going to be has no. It is deoxyribose sugar. The other side is going to be ribose sugar. Has nitrogenous bases, you tell us the nitrogenous bases. For the case of RNA, this one is going to be uracil. Then complementary bases, because the other one, this one is double stranded. Yes, this one goes with this. Then it means that RNA has no complementary bases, unless it's complementing with DNA. So we will see that when you are trying to translate it. It is a double molecule, it's a double helix. RNA doesn't form any helix, yes, because it's a short thing and it's a little bit straight. Yes, so basically that's the structure of DNA. What are some of the functions of DNA? What are some of the functions of DNA? So number one, sections of DNA forming genes carry hereditary information. So we see that DNA is very important in carrying the hereditary information. You look like your father because you obtain DNA from your father and your mother. You'll find out that the head looks like your father, the hands looks like your mother. So it means that these genes are being carried from one generation to another generation. What about when you resemble someone? You go to the village and then you find someone there, you resemble that person. Does it mean that something happened? Write in the comment below and then tell us what is your view concerning about that. Then when I see many comments, I'll come back and then I'll give you the reason why you resemble someone far. Think about it. All right, DNA consists of coded information for proteins. Number one, it carries inherited information. The way how you resemble, you obtain it from your parents. Number two, it consists of coded information for protein synthesis. So most our body is made up of proteins. The biggest percentage is proteins. So hormones, enzymes, which bring about the normal functioning of our body, so is made up of proteins. But it is DNA which codes for those proteins. We are going to look at protein synthesis when we continue. Yes. So, but it's DNA which codes for that. And I explained that in the previous lesson. So in other lessons to come, we will be explaining the protein synthesis. All right. So those are the two functions of DNA in our body. We saw that DNA has complementary base pairs. So let's look at the base calculations. In most cases, some books, they don't have this, but in most cases, we ask you this. The evidence of evolution. Number one is biogeography. As you hear the word, bio means life. Geography means distribution in different locations. So when you talk about, it means that you have life which has been distributed in different locations. So look at these camels. They have the same ancestor, but they have gone to different areas. When they went to different areas, this one here, this one there, this one there, this one there. So they modify. So this helps, or it brings about organisms to adapt differently. So if you look at them, you will know when and how did these species evolve. Why is it that you have different colors in humans? So you can use by geography because they are located in different environment. People from hot areas, you'll find out that they have different skin color. So that can help us to know how did these organisms evolve. Number two is fossil records. Fossils remains of living organisms. When you die, you become bones. So the bones will become remains. And if it happens that it takes for a long period of time and then other generation come and then they try to find your bones, and then maybe the generation is different, then they will study them and they say, oh, okay, in this time people who lived on this earth, they were like this, just using your remains so that they can now predict how life was by then. So that's what called the fossil records. If you look at this, it's sure that the, the layers of the earth, the lower layer will have the simplest form and the upper layer will have the more complex form. It shows evolution because increase in the complexity from simple to complex. So it means that the simpler one, they existed before, and then the more complex one, they existed just recently. So if you look at this horse, you find out that the skull you find in different strata of soil, different strata. Strata means layer in science. When you say stratum, it means layer. So when you say strata means layers. So different strata, you'll find out that simple will be in the lower and then the more complex one will be in the upper layers. So fossils remains have been found in rocks of all ages. Fossils of the simplest, where do you find them? You find them in the oldest rocks. Simplest, oldest rock. And then fossils of more complex organisms are found in newest rocks. You find them here, you see. And then you're saying that this supports the Darwin theory of evolution, which states that simple life forms gradually evolve into more complex ones. Like simple life forms, they evolve into a more complex one. That's why I said that you find out that you are more complex than your parents. Yes, you are more complex than your parents. It means that you evolve from simple to a more complex organism. You have another evidence which shows that evolution really took place, which you call the comparative anatomy. When you talk about comparative anatomy, this sometimes you call it modification by descent. You modify as you shift from one generation to the next generation. If you look at this deals with similarities and the differences in anatomy. Anatomy is the body structure. How do you, if you look at your body, how is the shape of it? Yes, if your shape is not the same as that of a cow, it's not the same as that of a pig. Yeah, they are not the same. But if you look at the anatomy, the inside part, they might have some similarities. So if they have some similarities, then it means that these organisms, at least they had some line of ancestry. So they have some common ancestor somewhere, but they diverged. They modified as or as different generation came up. So when you talk about organisms with the same ancestor have the same anatomical plan. This is what you call the pentadactyl limbs. Penta means 5, dactyl means big. It comes from the word digits. So if you look at your hand, you'll find out that you have 5 digits. 1, 2, 3, 4, 5. Your legs or your feet, they are 5. They are 5 toy. Or they are, yes. So if you look at them, they are 5. So if you look at a monkey, it has 5. So if you look at this picture, you will find out that you have human, you have 5 of them. 1, 2, 3, 4, 5. Yes. If you look at a cat, it has 5. 1, 2, 3. They have 5. If you look at a whale, yes. If you look at it outside, it is different. But if you look at its anatomy, that's why we say that we compare the anatomical plan or structure. If you look at its anatomy inside, you'll find out that it also has 5. 1, 2, 3, 4, 5. If you look at a cow, it also has 5. But some of them are fused. If you look at a chicken, it has a 5. You see them 3, 1, 2, 3. in front, another one is behind, making them four, another one is somewhere here on top. Yes, so there are 5. So all those, it shows that at least maybe we have some line of ancestry. So it means that we shared an ancestor at a certain time t. So if we study this, it helps us to know who came from who and who diverted from who. So it shows that evolution took place. If you look at this bed, it's also, it has five of them if you count them. So you can compare many animals, most especially the vertebrates. Most of them, they have almost the same anatomical plan, which shows that vertebrates came from the same ancestor, but we try to divert to different areas. You go to different areas. That's why you call it divergent evolution. You start with the same ancestor, but you go to different areas, and then you modify yourself. Get an example. If it is a cat, it is modified to capture a prey. If you look at a whale, it is modified into swimming. If you look at a bat, it is modified to fly. You understand? So it means that if you look at these structures, it shows that evolution took place. You also try to do the same. If you go in water, you will find out that you want to walk the same way you walk on land. You try to imitate the fish which are found in water. If you go to air, you try to fly like a bat. So basically, that's how what you call the comparative anatomy looks like. It means that all vertebrates we share a common ancestor, but we went to different environment and then we try to modify according to where we went to. And then if you have almost the same structure, then it means that you almost share the same environment where you are staying. So another evidence is genetic evidence. When you talk about genetic evidence, you're talking about the genes. So we are saying that one of the strongest evidence for common descent come from the gene sequence. Like how? If you have the genes which are almost the same, then it means that we share a common ancestor. Or the line of descent is just recent. It means that you just diverged from each other just recently. But if you have different genes, the percentage of similarity is too small, then it means that you diverged long time ago. So you're saying that organisms are likely to have common ancestor if they have, number one, identical DNA structure. You find out that chimpanzees, they share almost 98% of DNA with humans. Then it means that we are related to chimpanzees. So similar sequence of genes, it means that the way how they are sequenced, it means that the way how they are aligned. So if you have the same sequence, then it means that we share a common ancestor. Similar portion of DNA with no functions. We have what you call the nonsense DNA. Nonsense DNA is the part of DNA which doesn't have any function. It's just there, but it can be mutated. When it is mutated, then it means that it can have a function in those organisms where that mutation has taken place. But the biggest percentage of DNA has no function. And then you're saying that similar mutations, we have some similar mutations. We will see this when you are talking about the out of Africa hypothesis, that human beings originated from East Africa. So East Africa is the mother. Then if you look at this picture shows the different genes which are in different organisms and the differences. This shows the differences, the percentage in differences of the gene. Humans with another human, a human with another human, they have 0% difference in the genes in terms of the hemoglobin. And then humans with the chimpanzee, they have 1% difference. Then it means that, oh, humans and chimpanzee, we are almost the same. And then humans and monkeys, they have 8%. It means that, yes, we are almost the same, but the monkey is a little bit different from humans compared to the chimpanzee. And then rats, we have 27%. It means that one day we and rats were the same organisms, but we diverged long time, and then it created 27%. What about humans and pigs? I've not talked about them, and then they have a very big similarity. So it means that you guys, when you are eating, make sure that you eat what is supposed to be eaten. No, don't eat your brothers and sisters. Okay, we are saying that you have frogs. Frogs, we are two different, 67%. Chicken, two different, 45, is still big. You can eat, it's fine. But chimpanzee, you can eat chimpanzee. You're eating, this is a real problem. So, but in the Chinese, they told me that in Chinese, gorillas, all those things, they eat. So basically, that is genetic evidence. It shows that if you have the similarities, then it means that you have lines of descent. Is it clear? By the way, did you know that humans? Let me tell you something. This is a little bit different. Did you know that humans and the monkeys, they're almost the same people, the same organism? Yes, you share 8% difference. No, 92% similarities and 8% difference. Did you know that humans have blood group D, which you call the rhesus blood group? That's why they say that your blood group A positive, blood group B positive, or blood group B negative. Means that if your blood group A positive, I don't mean HIV, no, I mean blood group A positive, you have a rhesus blood group, which called blood group D, which was discovered in a rhesus monkey. That's why it's called. People who are not producing, like you marry someone and then you keep on having miscarriages. Okay, that's genetics. Though it's a part of genetics, but I'm not going to talk about that. But that is genetics. If we go back to genetics, you will ask about that. I'll explain how this happened and how you can solve that. But it shows that if humans have blood group D, which is rhesus blood group, and then also monkey has it means that they have the blood group of the monkeys called the rhesus monkeys. Don't you see that we are almost the same? So those are our brothers and sisters. Another one is saying that comparative embryology. Here you are describing, or we are comparing the embryo of different organisms. Yeah, you guys, if you look at, let me just bring it before even I start talking about it. Here you have different embryos. If I cover this part, you won't be able to tell me that this is human, this is a rabbit, this is a cow, this is a pig. You see that you and the pig, you're almost the same. You and the calf, you're almost the same. You and the rabbit, you're almost the same. So it means that we have a common ancestor. One scientist, American scientist, said that ontogeny recapitulates phylogeny, which means that history repeats itself. When you say ontogeny recapitulates phylogeny, in English, we say that history repeats itself. What does it mean in actual sense? What does it mean? It means that during our life development, we repeat our evolutionary trends. So you keep on repeating what you went through during evolution. That's why we are saying that at a certain time T, you had a tail. That's why when you are developing, you have a tail. At a certain time T, you stayed in water. That's why you have gills. And gills, we use them for gills are used by organisms which are found in water. That's why when you go, you drown in water, you cannot stay there alive. Why? Because you don't have gills. The gills have been modified into lungs, but the fish will stay there. You understand? So it means that if you have a gill in your developmental stages, then it means that at one time T, you had gills. So now we are saying that the study of the development of anatomy of organisms to suit to its adult form provide evidence for evolution as embryo formation in widely divergent groups of organisms tend to be conserved. It means that you keep on, or you conserve those stages you went through during evolution. For example, are the vestigial structures such as tails or gills. Vestigial means those structures which had a function before, but they are no longer there. So it shows that at a certain time t, you had the tails. So because the tail is not important to us, we call it vestigial. So it decreased, it disappeared. Vestigial structures. So the tail is a vestigial structure. Yes. Gills, we no longer having gills, they modify into lungs. In human can be found in embryos early stage of development. So because we share or we look like other organisms, chicken, tortoise, salamander, fish, it means that all these organisms had a same ancestor at a certain time t, but we evolved into different species. Another evidence is cultural evidence. This is basically linked to humans. It shows how humans used to have to use these tools. When you talk about, we will see in human evolution, we have different stages. We have the Homo habilis, and then Homo erectus. When you talk about Homo habilis in English, it means that handyman, the people who used to like using tool, using fire. So this is the period. So if you find this in South Africa, then it means that those organisms or those humans were staying in South Africa. And then we can calculate how many years ago did those organisms which used those tools existed. So if you do the line of, or we link them, we can see that evolution took place. And then we see that these were humans, but they try to evolve to a more advanced organisms, which we call Homo sapiens.
Welcome back to our class. Don't forget to subscribe if it is your first time to visit our channel. Evolution in present time. What happens? Does evolution takes place? We want to see it because we are saying present time. Then it means that evolution is taking place. Let's look at it in detail. Describe how mosquitoes develop resistance to DDT. How did it, like when you apply DDT, it does not affect mosquitoes anymore. Then it means that there is a resistance which has been which has been formed. So we need to know that. So that is the evolution. It means that evolution, remember I told you that evolution is increasing the complexity of an organism. The organism become more complex. If the organism become more complex, it means that what was working on it, it's no longer working on it. It has become more complex. It has modified its structure. Then it means that it cannot It cannot be affected by such. So you're saying that now you have these mosquitoes, yes? If the mosquitoes, you have this kind, you have this kind, you have this kind. So what happens when you apply the first application of DDT? Yeah, you see that all these ones, they die. Yes, you see them that they have died. Only this and this, they have survived. So it means that there is variation among the species. There is variation among the species of mosquitoes. Yes, during the first application of DDT, yes, those which are resistant will survive, while those which are not resistant will die out. Those which are resistant will reproduce in large numbers and they will pass their genes to the next generation. Yes, when they pass their genes in the next generation, they reproduce in large numbers. And then when you apply again DDT, it means that it cannot affect them. So it means that now the mosquitoes have become resistant to what? To DDT. So first application, there is variation among these mosquitoes whereby some are resistant and some are non-resistant. But the one which are non-resistant are very many compared to those are resistant. So when you apply many which are non-resistant, they're going to die. You see them, they're fading out. While those which are resistant will survive. Yes, these ones which are resistant will pass their genes to the next generation, will produce in large numbers and pass their genes to the next generation. The second application, yes, of DDT, yes, they will not be able to affect them. Why? Because the one which are resistant have survived. Then it means that the DDT will become ineffective to the mosquito. It happens in when you take drugs and you don't complete the dose. It happens HIV, it happens in bacteria. Yes, it happens in their life. So let's look at it. There's a large number of degree of variation in the mosquito. Yes, we saw it. When the DDT was first used, it killed off the large number of mosquitoes, but some mosquitoes were resistant to the DDT and they survived. Those which are resistant, the black one, you saw it, they were resistant, so they survived. When they survived, they passed. Those that survived were able to reproduce. They reproduce in large numbers, increasing the population of resistant mosquitoes. Yes, continued use of DDT had little effect on the resistant mosquitoes. It means that those which are resistant, now they have conquered the environment, so DDT will not be working on them. So at the end of the day, you'll find out that DDT will become ineffective to mosquitoes, or it becomes effective in killing the mosquitoes. Hence, we are saying that hence the resistant mosquitoes increased, and then non-resistant mosquitoes, they died or they decreased in numbers. So basically, that's it. That is the present day evolution. Other examples which can help you, you can do it with your teacher. Number one, this is resistance in bacteria. Yes, development of resistant strain of tuberculosis, that is bacteria, Mycobacterium tuberculosis, due to the mutation. So this the bacteria, it will reproduce in large numbers. As it is producing large numbers, it produces some resistant. When you apply the antibiotics, yes, these ones which are not resistant will die out, and those which you see they have died out. Those which are resistant, the two of them, they will survive, and then they will reproduce in large numbers. They will pass their genes to the next generation. After many generations, or after successful application of the antibiotics, yes, the antibiotic won't be able to work on the bacteria, and then it means that the resistant one will survive. That's why when they are giving you medication, sometimes they give you a combination of medication, because they know that there could be some resistance, so they must give you the medication in different versions, so that even the resistant ones, they can be resistant to this drug, but they might not be resistant to another drug. So they give you multiple drugs. But what other people do, they just take one, and then when they feel fine, they stop taking medication. You are just creating monsters in your body. You have to take the drug completely without leaving them, the way how the doctor has told you to do. So even if you're fine, you have to complete the dose, so that you kill this resistant and non-resistant completely out of your body. Then another example is HIV resistance to antiretrovirus, ARV, antiretroviral, and antiretroviral, that is ARV, antiretroviral medications. So you see that if this is the HIV, what happens? You have some resistant and some non-resistant, some resistant and non-resistant. When you apply the first medication, the resistant ones will survive, will keep on surviving, while the non-resistant will die. You see that the red ones are dying. And then after many generations, the non-resistant ones will die out, and then the resistance will survive until the end of the day. You pack. So it means that now that medication of HIV cannot work on you anymore. Yes, it cannot help you out. It means that you have to go back and then change the medication. That's how present day evolution is happening. Present day evolution, you can also have another one in black peppered moth, and then the white peppered moth, industrial Mendelian, the black peppered moth, and then the white peppered moth. So you can have also such examples. So basically that's it. And then human evolution is switching in. Then I will see you there when you come to human evolution. Don't forget to subscribe for our new videos.
Today we are going to be looking at the three lines of evidence used for common ancestor of all hominids. The three lines of evidence used: number one, fossil evidence. Fossil evidence, we are going to look at the different fossils. Remember, fossils are remains of living organisms. The organisms which existed, their remains form what called fossils. Number two, we are going to be looking at the genetic evidence. In this case, we're going to be focusing on the mitochondrial DNA. And then number three is cultural evidence. So let's look at the fossil evidence. This one is the one whereby students, they think that is the toughest part of human evolution, but it's not tough. It's only that you just need to understand the concepts, and then you will be able to pass exams concerning about this. I'll show you even how do they ask questions concerning about this fossil evidence. So we are saying that under fossil evidence, you need to understand what is a transitional fossil or what is a transitional species. A transitional species is the one that shows an intermediate characteristics between two genera. Remember, when you say genera, we are talking about a genus. If it's one, if there are many, we call them genera, or different species, or between two species. Or we can define a transitional fossil or species as is the one with the characteristics common to both ancestor species and the species that follows. That's what I've tried to explain, that at least you have to be with the same characteristics. Look at yourself. You and your father and your grandfather or grandmother. You and your granny could be the different species. For example, if your father has some characteristics from the granny, and he also has some characteristics from you, then we can say that a dad is a transitional fossil or a transitional species. It has the characteristics of the granny and has the characteristics of the son. So it means that is having two characteristics. So a transitional species is the one which has both characteristics of the ancestor and then the characteristics of the following what species. Yes. So here is an example of a transitional fossil. You have this animal, and then you have this animal. Yes, they are birds, dinosaurs, whatsoever. They are animals. They are under kingdom Animalia. So you have this animal, and then you have this animal. So there is a transitional fossil of species here. How is it a transitional species? If you look at this species, there are some characteristics of this bird or this animal, and then some characteristics of this bird or this animal. So if you look at these feet, you'll find out that they are looking like this ancestor. Yes. If you look at this part of the wing, it looks like the ancestor. If you look at this part, it looks like this one. If you look at this part, it looks like this one. So it has both characteristics of the ancestor and the characteristics of the following. So we call this we call it a transitional species or a transitional fossil. What are some of the important sites in Africa which support this fossil evidence? Yes. Number one, we are saying that the first, we have countries. We have South Africa. We're going to look at Kenya. We're going to look at Tanzania. We look at Ethiopia. We're going to look at Chad. If you look at this map, it's showing you the different countries in their location. So let's look at the evidence, the fossil evidence. Here you need to know at least the scientific name, common name, who discovered, where it was discovered, and at least the age. So in this case, I've not included here because I have already designed a small clip which has all that information. So you have the scientific name. The first fossil you need to understand is Adepithecus ramidus, which we call Ad. Yes, who discovered it is Tim White. Yes, and then where exactly is in Ethiopia. Sometimes this one becomes the common name. Sometimes when you're asking, we might not ask you whether the common name or the scientific name. We just ask you a question which is open, whereby you are supposed to answer depending on the question. If it requires scientific name, answer in the scientific name. If it requires a common name, answer the common name. If it's open, you can choose whatever you want. So we have the Australopithecus afarensis. This one is called Lucy. The person who discovered it is Johansson, Johansson discovered it in Ethiopia. Lucy, those people who watched a movie called Lucy, you can even see how did life try to evolve. We have the three fossils. We call them Australopithecus africanus. The first one is Taum child, who discovered it. Raymond Dart. Where exactly in Taum? It's called Taum child because it was discovered in Taum. Then we have Mrs. Place. Mrs. Place, who discovered Robert Bloom, discovered in Stackfontein. Yes, in Stackfontein caves. Sometimes we call them Stackfontein caves in Taum. And then you have the Littlefoot. Littlefoot was discovered by Carl in Stackfontein. Still, they discovered it in the same place like Mrs. Place. But these they didn't discover them in the same period of time. So sometimes you can ask a question that give two Australopithecus africanus species are found in Africa or found in South Africa. Then you can now name Townchild, Mrs. Place, and then Littlefoot. Sometimes you can ask which one of the following is non-Australopithecus africanus species. Then you say that Townchild, Mrs. Place, Littlefoot, and then Karabo. Because Karabo is also Australopithecus, but doesn't mean that it is Australopithecus africanus. Sometimes you can ask a question, just give three Australopithecus species you know. Here, when you say Australopithecus species, we are not saying that give the Australopithecus africanus. It's just Australopithecus. Then it means that even Karabo can be what? Included. But if they are specific to africanus, then you have to answer only question concerning about Australopithecus africanus. Yes. So we have Australopithecus sediba. This one is called Karabo, who discovered it. Is Lee Berger, and then where exactly in Malapa in Corridor of Humankind in South Africa. Yes, Karabo has some unique features. Sometimes it's called a transitional fossil, or a missing link between Australopithecus, and Homo habilis. So this is a missing link between Australopithecus africanus and Homo habilis, or a transitional fossil. I explained the transitional fossil that is a species which has some characters of the ancestor and some characters of the next species of the following species. Australopithecus sediba has some characteristics of Australopithecus africanus and Homo habilis. Therefore, it is regarded as a missing link. Then another one is Homo habilis. Homo habilis means the handyman. Homo means man. Then habilis means handy. So who discovered it is Mary and Louis in the Olduvai Gorge. They discovered it in Olduvai Gorge, Tanzania. So basically, yes. And then we have Homo erectus. Homo erectus. Erectus, upright. Yes. It means that upright walking man. Yes. Homo means a man. Yes. A man. Yes. Homo habilis. Yes. Man. Yes. So it means that upright walking man. Upright man. Yes. This one is the one who discovered it, Du Bois, in Indonesia. So we have even in other areas, but the oldest was discovered there. Then Homo sapiens is called the wise man. Wise man, yes. I heard of a school called wise man. Yes. So maybe we can call it Homo sapiens school. All right, wise man. Yes, who discovered is Tim White, still again. He discovered this in Limpopo, in Western Cape, in KZN. Yes, all those places they discovered those Homo sapiens. Now, as I'm asking you, are you not Homo sapien? How comes that they're telling us that they discovered Homo sapien? And even the one who discovered Homo sapien is a Homo sapien. Does it make sense? The person who discovered Homo sapien is a Homo sapien. So he discovered himself. All right. Here they are trying to mean that the oldest Homo sapien. Yes. Oldest Homo sapien. However, the person who is discovering is a Homo sapien, but he discovered the oldest Homo sapien. And where did he discover it from? Discovered it from those areas. Then note, Australopithecus sediba, which we call Karabo, is a transitional fossil. I explained this. That is a transitional fossil between Australopithecus africanus and the Homo habilis, the handyman. Between this and this, between this and this. So this Australopithecus sediba is the one which tries to link between these two, and sometimes we call it a missing link. a missing link, a missing link. Yes, number 2 is genetic evidence. Genetic evidence. This is mitochondrial DNA. Mitochondrial DNA. Mitochondrial DNA. Remember, when you're talking about mitochondria, we say that mitochondria are semi-autonomous. They are self-reliant. They have their own DNA. They are not being controlled fully by the DNA in the nucleus. No, it has its own DNA. However, the mutations which occur in the mitochondrial DNA, they are rare. And if they are there, we can use them to trace our lines of descent. But there is something special with the mitochondrial DNA. Yes, let me give you this example. Remember, when we are talking about fertilization in reproduction, we say that the sperm, when it fuses with the ovum, only the head of the sperm fuses with the ovum. The tail does not enter into the ovum. And remember, the middle piece and the tail, the middle piece is the one which has the mitochondria. So it's only the head. So the middle piece and the tail, they don't enter into the egg. What happens then if it is like that? Then it means that the egg will only have the mitochondria of the egg or of the ovum. So all of us, we have the mitochondria from the mothers, from our maternal parents. So it means that if there is a mutation in the DNA of the mitochondria, it means that only that of the lady will be passed to the next generation. So if we try to trace that DNA, we can see that, okay, it came up like this until we find where it originated from. So I want to trace where the mitochondria from this. So I have the person here. I go like this. Father, mother, yes. In this case, I don't go. Why? Because it won't give me. I'll only go with the lady. Yes, you see. Now, when I reach here, the father does not give me the DNA. No. So I'll go with the female. I continue. When I reach here, then I continue with this. It means that the father doesn't give me. So this is where the mutation maybe happened. Or I can continue until I find the origin. So we are saying that mitochondrial DNA or mitochondrial DNA is inherited only from the maternal line, the mothers. And then you're saying that analysis of mutation on this mitochondrial DNA, yes, shows that the oldest female ancestor were located in Africa. The scientists say that Eve, Muslims call her Hawa. Yes, Eve originated from Africa. So Adam and Eve, even for guys, is East Africa. So it means that we are the owners of this world, because Hawa or Eve and Adam originated from Africa. That's what they say, scientists. So we are saying that, and then all humans descended from her. That is the mitochondria of Eve. That's the one I've been talking about. Yes. So basically, that's how you can explain. Whenever you see a tick, it means that, guys, don't miss out that point. Don't go to exam without knowing these evidences. I repeat, don't go to the exam without knowing these evidences, because if you don't know them, then it means that you won't be able to get the marks. All right, let's continue. Then you are saying that cultural evidence, tool making. This one, using stone tools with cutting and sewing, improve the human nutrition. Like if you can cut the meat, so it improves the nutrition, so that you can kill the animal, so that you can obtain nutrients from this animal. Development of stone tools became more sophisticated as hominins evolved. As these hominins are evolving, yes, so the tools became more advanced. It's like a phone. 2000, the phone was just like that, but as you improve, you go on going towards 2020, it becomes more sophisticated. So that's what happened during those ages. And then you're saying that 2 required planning and thought. So it means that also the brain was also developing, because they require planning and thought. You don't just come from nowhere and then you develop a tool. No, you can see them as they are just light, you can design them, but if you were the one by that time, you wouldn't have done that. A generation which will come, they will laugh at us, the kind of phones, the kind of computers we are using. Yes, they will laugh at us. You understand? Yes. So it means that technology or development of brain keeps on evolving and evolving. So here is the tool they started with. This check the kind of tool they started with, and then they started to modify it, and then they modified it up to this level. It shows that there is development in thinking. There is development in thinking. So this shows that from here to this point, it shows that there is an increase in the brain. You can see that the kind of tools they are using here, even the brain size here, you see is just very small. The kind of tools that are here, these ones are more advanced than this. Even the brain size bigger than this one. This is Homo habilis, this is Homo erectus, and then the kind of tools now we are using are these ones. This is the Homo sapiens. Yes. So by then we used to have axe from stones and pangas from the stones, knives from stones, but now we just have to modify them. Even now we're using the stones, yes, but they are just modified to suit the requirements. Yes, basically those are the evidences out of Africa hypothesis, and the evidence for African origin of modern humans.
Welcome back to our class. Don't forget to subscribe if it is your first time to visit our channel.
They're saying is a cross which involves only one characteristic or trait. Trait is the same as the character, and it's being shown in the genetic cross. This guy Mendel put forward the law of segregation. He said segregate means that now individuals during meiosis, they are supposed to segregate, they are supposed to divide, so that the offspring must receive one from the parent male and one from the parent a female. So how do they form these gametes which are being formed, which are being given to the offspring? They're being formed during the process of segregation. So he says that diploid, it means that the organism has 2 sets of organisms. For example, how many chromosomes do you have? You have 46, but you have 23 pairs. So if there are 23 pairs, then we say that you are a diploid or diploid. Randomly, you don't have a choice which one is going, which one is not going. It's randomly to the offspring, such that the offspring receives one allele from each parent. The characteristics you have now, the parent gave you, each parent brought one allele. If you are tall, for example, it means that those genes you have, one parent gave you one allele for tallness, and another parent gave you one allele for tallness. So if both of them, they gave you a dominant allele, then it means that you are homozygous tall. If one gave you a dominant allele and another gave you a recessive allele, then it means that you are heterozygous tall. So that is the meaning, that each parent is supposed to give you one character. Let's look at how do we mark. This is how we mark in exam. And if you miss something, then it means that you won't get a mark. Number 1, whenever they give you a genetic cross, this is what you are supposed to write. P1, which means parent 1, phenotype. Phenotype, physical expression of an organism. We get this from the question. A man who is dark-skinned marries a lady who is light-skinned. Dark-skinned man, light-skinned lady. That's it. That's the phenotype. So you go ready. Then genotype. Now, who is this dark-skinned, light-skinned? Is that person homozygous or is heterozygous? That is the genotype. So when you read the story they have given you, you'll find out that you'll be able to scoop out what is called the genotype. So we give you a tick here, we give you a tick here. From there, because you have now the genotypes, you have the genes for each parent, then meiosis must take place, segregation must take place. When it takes place, yes, you form what you call gametes. And then when these gametes, now you have, for example, the sperms this side, you have the ova this side, or the ovum this side, then fertilization must take place. Fertilization must take place so that offspring can be formed. Then you have F1, which means first filial generation. And then now you get the phenotype of the F1. I'm going to explain this. After that, you need to get what called the ratio, or the genotypic ratio, or the phenotypic ratio. Then you use the genotype to obtain the phenotype. Use the genotype, description of an organisms in terms of genes, and then after that you'll be able to get what you call the phenotype. Writing the word meiosis and fertilization will also give you a tick. If you don't write it, you lose that tick. So now you have the gametes. These ones you bring them here, and then these ones you bring them here, or you can swap them. So that's how we do mark. How do we mark genetic cross? That's how I've been trying to explain. This is a practical example. So P1, F1, we give you a tick. Meiosis and fertilization, we give you a tick. Writing the phenotype, we give you a tick. Writing the genotype, we give you a tick. Then crossing them, putting the gametes here, and then you cross them correctly, we give you a tick. Writing the phenotype, the genotype, we give you a tick, and then writing the phenotype, we give you a tick. P1 and F1 can change to P2 and F2 if it is the second generation. So now that's it. Another thing which we also do when we are marking you, yes, we make sure that there is a compulsory mark in the genetic cross. If they ask you a question, maybe P1 and F1, it should be the compulsory mark. And you have, this question is how many marks? For example, there are 6 marks. And then if you count here, 1, 2, 3, 4, 5, 6, 7, there are 7 marks. And then they say that P1, F1 is the compulsory mark. So you get the rest of these marks. If you count them, there are 6. We won't give you 6. We will give you 5. Why? Because you did not get this one correct. Maybe you only wrote P1 and you didn't write F1. So you didn't get this one correct. So when you are counting, we first count the compulsory mark and we count the rest. So make sure that when you are crossing it, at least cross it and maximize all the marks. Okay, let's look at dominance, which are the types of dominance. We can go through them very fast. Walking on 2 limbs. So that is what called bipedalism. What are some of the factors which causes bipedalism? These are the factors. 1, 2, 3. Those are the 3 factors which cause bipedalism. Number 1, foramen magnum. The position of foramen magnum, it will show whether it is bipedal or quadrupedal. If there are 4, walking on 4, it's quadrupedal. Or yes. And then you're saying that it moves to a more forward position. If you are bipedal, it means that it goes towards the forward position. This will help in allowing the spinal cord to enter vertically. You saw from that diagram above that the spinal cord enters direct into the head. So it makes the person to walk upright. And then pelvic girdle, we call this one pelvic girdle. Yes, it is short and wider or broad. So if you look at it, it is short and then it is wide. I'm going to explain this one more. Yes, why is it short and wider? It is to support the upper body weight. Guys, you need to know these factors. We love them. We love them. And then you are saying that the spine, this is the spine, this red color, the spine, how about it? It's more curved. So if it's more curved, it means that it can allow the compression. Look at you when you jump from a high point to a low point. You don't just jump and stand straight. No, what you do is you jump and you try to squat, and then you stand again, just to make it to have that compression. So the reason why this one is like this is just to have the absorption of this pressure from up, so that now you can stand upright. Yes, so to absorb the shock or to allow flexibility during movement, or to allow support. So all that you just choose one, and then you'll be able to get there. So factors that affect that caused bipedalism: foramen magnum, position of foramen magnum, you have to tell us. Pelvic girdle, you have to tell us where is it. And then at the spine, you have to tell us. We said bipedalism is walking on two limbs. So position, that's the position you're talking about. And then short and wider is this part, which must be short and wider. And then this one is this spine you see in this direction. So let's look at this difference between bipedalism and quadrupedalism. Yes. If you are walking on two limbs, what will happen to the position of foramen magnum? Foramen magnum is forward. It is forward. It is going this side. From this point to this point, you see the distance is big. While this side, the foramen magnum is backward. Here, you see it is, this from here to here is very small. So it is behind. It is backward. We don't say behind. We say backward. I repeat. Please don't use your own English here. We want these words. That's what we want, so that you can score a tick. Remember, we use what you call KO, knockout, so that the examiner does not have any issue with you when he's giving you a tick. Thank you. Number two is more curved spine. More curved spine. If you look at it, the spine is curved. You see it? I explained it. So it's S shape. Yes, just we say that to allow the compression from top. Yes. While this one is C shape. So you see that it looks like a C, while this one is S shape. So less curved spine. Less curved spine. Sometimes you can say C-shaped spine. You can say more curved spine, or you can say S-shaped spine. So you don't need to write two, just write one. Then you get a tick. So they're saying that now these are pelvic girdles. You see that this one is short and wider. Always short individuals are bold. Yeah, bold and wider. Always all right. Okay, let's continue. Then you are saying that this one is long and narrow, is small here. All right, we're saying that short and wide, while this one is long and narrow. This is what I'm talking about. This is shortness, while this is the wideness. This one, this is the longness, while this is the narrowness. So that's how you can interpret it. If you look at it, we will bring just skull without label, and then we ask you to give us the differences. Just look at them and which one supports the bipedalism, which one supports the quadrupedalism, and give the reason for your answer. Yes. So yes, we will see some questions. So what are some of the advantages of bipedalism? We can bring this in a 20-marker question. Yes. So you're saying that free hands to pick and they carry. When you are walking, when you're giving examples, when you're giving answers, you must compare to yourself. If you compare to yourself, then you'll be able to answer such questions. So free hands to pick or carry food, to use tools, and then handle weapons. So when the hands are free, you can carry food, you can doing multiple tasks. You can be multiple tasks. And then number two, standing upright. Why is it when you stand upright? What is it for? Yes, it gives us a wide view. You can see someone far. That's the problem of being short now. If you are short, you can't see someone far. Oh, but yeah, the also short people have advantages. Sorry if you are short. I'm not trying to say that it's bad. All right, number 3 is movement. It gives us movement. How about movement? It makes it easier for us to move, and then we use less energy. Imagine when you are walking on 4 limbs, the hands, the energy you try to use, it is too much. Then another, reduce the body is exposure to sunlight. Then another one, it rises the body away from the hot ground. That's what I've also talked about. So if you are, imagine when you are sitting, you are sleeping, the amount of sun which will be on you will be more than the amount of sun when you are standing. And then if you are on the ground, the amount of heat which will come into you will be more than the amount of heat if you are far from the ground. And then another one, 6 organs can be exposed. We can see them. Oh, yeah. You see that you can see a lady a little bit far. Say, oh, these are the boobs. Yeah, these are the kind of boobs I'm looking for. 6 organs can be exposed nicely, and then we know that, okay, we either go for courtship or you don't go for courtship. So being bipedal is a good or is an advantage. Guys, try to understand these points. Here are some of the questions. Number 1, tabulate the observable difference between skull 1 and skull 2. Skull 1, skull 2, skull 1, skull 2. Observable. What do you see? So let's see. What do we see? Do we see foramen magnum? No, I can't see foramen magnum. You don't talk about it. Do I see the broadridge? Yes, broadridge here are pronounced, are more pronounced. Here we have no broadridge or less broadridge. Here they are more pronounced. Here they are less pronounced. That's number 1. Number 2, sloping face. Sloping face here. Here is flat face. You see, you can see what you see. I see the canine here. The canines here are well developed. Yes, while the canines here are less developed. So you can see this one is more prognathous. The jaws are protruding out, are protruding out. While here the jaws are not protruding. You know that some people are like this. You mean that you are prognathous. A little bit primitive. Primitive means that, yeah, you are still in 1 million years ago. For us, we are in 1 million years ahead. Sorry if you are one. All right. Then you're saying that. So those are some of the differences. But what you see, yes. Then you're saying that number 2, give 4 characteristics of the upper limb that humans share with other primates. Upper limb, only upper limb. Opposable thumbs, yes. Flat nails instead of claws. Yes, you can talk about free rotating arms. Upper limb only. Don't tell me sexual dimorphism. No. They are not talking about that. They are talking about upper limb. Guys, make sure that you read the question nicely. So you have almost 66 marks there. Mahala. Ish. Guys, you have to get this mahala thing. They're saying that explain how an increase in brain or cranium volume is related to intelligence. We say that increase in the cranium, it means that it will accommodate more brain. It will have more brain cells. I tried to explain this, and this is an indication of intelligence. A bigger cranium indicates more brain cells, or a big brain, which indicates a greater intelligence. Humans are bipedal organisms. Yes, I see that humans are bipedal. They walk on 2 limbs. What is meant by pedalism? Bipedalism, walking on 2 limbs. Simple. Go. Or walking on 2 legs. Walking on 2 legs. Yes. Explain how each of the following skeleton structure have contributed to bipedalism. Number 1, foramen magnum. I told you, forward position. Why is it for? Is to allow the spine connect direct to the skull. Yes. Pelvic girdle, short and wider. What is it for? To provide a surface area for upper weight. It can support the upper weight. Spine is S-shape. We talked about this. What is it for? S-shape. It allows the compression. It absorbs the compression. Yes. So that now the organism can stand upright. So you guys, we are going to come back. We are going to come back with this evidences or using. Now we are going to force it. our brain, if you look at in the picture, you see that it is bigger compared to this. What is inside here is what makes the human to be reasoning more than any other animal. Let's draw a line here. You see the line here, and then draw a lower line. You will find out that all this is human brain, but for this one is very small. You see now here, all this is human brain, but this one is very small brain. So it means that human have a bigger brain compared to other primates or other or apes. So we have a bigger brain. So this one is large, the other one is small. So we can say that cranium is a part of a skull which encloses the brain. So we have been told, you know about the cranium is this part which encloses this bone which encloses the what? The brain. So humans have a larger brain, more brain cells. It means that we can reason. So that makes it, yes, to reason, to have a greater intelligence. So they have more brain cells to the body size, This is an example of the elephant I gave you, that although the elephant has a bigger brain compared to us, but we have a bigger brain compared to the body size. So which suggests that we are of a great intelligence. And then we are seeing here that human skull, if you look at it in detail, yes, these are the things we are going to ask you on the human skull. So they're going to ask you, we have a bigger brain, a large brain, small brain. The face is flat here. The face here is sloping. You see them? The face is sloping. Here, the canines are reduced. We don't have well-developed canines. These ones, they have a well-developed canine. You can see them here. And then if you look at the foramen magnum is that hole. Is that hole, this hole, which allow this spinal cord to enter. So if you look at here, it is forward. It's forward to the side of the mouth. It's forward. So we say that foramen magnum is forward. That's how we want it. If you change it, that is in the center, is no. We won't give you a tick. We want you to say that foramen magnum is forward, or it is in the forward position. And then here, foramen magnum is you see is running backward. So it's running backward. So how do you write it? You say that foramen magnum is backwards. That's what we want. You change the statement, we give you 0. Yes. So basically, that's it. This one helps us, the foramen magnum, this hole, it helps us to know whether the individual is walking on 2 limbs or is walking on 4 limbs. If he's walking on 2 limbs, we say this bipedal. So we say that that's bipedalism. And then this one has a U shape. Yes, if you look at it, it is a U shape, while this one is rectangular shape. If you draw a line here, you see it is rectangular. Rectangle, is a rectangular shape. This one is a U shape. Sometimes they call it C shape. Yes. So basically, you need to know those things. If you look at this part, this part is almost flat, while this one is protruding out. So we will say that it has a well-developed broad ridge. This is the broad ridge they are talking about. Broad ridge are well-developed here. Here we do not have. So basically, you need to know those differences. So in exam, we will not bring it like this. This is so beautiful. Now in exam, we will bring it like this. This one will look like this, while this one will look like this. And then this one will look like this. So you see that the teeth are out here. So when we are bringing exam, we don't bring you like this. No. We bring you like, so we bring you like this. And then this one will look like this. That's how, then you ask you, we want even label. We'll say that give the differences between skull A and skull B. And then from there, we will know that this one broad ridge will develop, this one will develop canines. We'll talk about that. So I'll show you how you can answer this question. And then you're saying, what are the differences between the apes and humans? So I'm going to repeat, but now here we are talking about other things we didn't talk about. Large cranium. The cranium here is large, while this one, the cranium is small. We saw that the skull of where the brain goes, for the case of humans is large, this one is smaller. And then this one, the face is flat. Yes, the face is flat. While this one, the face is sloping. This one, the brow ridge are not pronounced. I showed you on the face that the brow ridge are not pronounced. While these ones, the brow ridge are pronounced, or they are well developed. Canines here, we saw that they are reduced, so they are small, while these ones canines are large, or the canines are well developed. And then these ones are protruding. The jaws, if you look at the jaws, let me go back. If you look at these jaws, the jaws are protruding out. Even you can see it here. But here the jaws are not protruding out. Yes, you see that the jaws are not protruding out. But these ones, they protrude out. So we are saying that jaws are less protruding. They are less protruding. We can say that is less prognathous. Less prognathous. And then this one more protruding. We can say that it is more prognathous. That's how we can talk about. Then you're saying that this one, foramen magnum, is forward. I repeat, foramen magnum is forward, while this one, foramen magnum, is backward. So we say that this one helps us in determining whether the animal is walking on 2 limbs or is walking on 4 limbs. And then we are saying that more curved spine, we are going to look at this one, more curved spine, that is S shape, while this one, the less curved spine, we call it C shape spine. I'm going to explain this one in detail. Yes, and then short and wider, that is the pelvis. The pelvis is short and wider, while this one, the pelvis is long and narrow. I'm going to explain this and this more. And then we are saying that small and semicircular pallets, while this one is long and rectangular pallets. So these ones are small. For the case of humans, if you look at your jaw, yes, they are small and circular. You see, I showed you that it looks like C. It's a C shape, while this one is rectangular. If you look at it, you saw that it's like a rectangle. Yes, while this one is a C, or it looks like a U, depending on the direction where you are facing. Yes. So it's like 6 and 9, almost the same, depending on the direction. So it could be C or it could be U shape. So what is bipedalism? Bipedalism is walking on 2 limbs. So if you walk on 2 limbs, it means that human evolution. These are the things which we need to cover in this human evolution. Number 1, evidences of common ancestors. So we are going to look at them and then see how we can answer questions concerning about that. And then out of Africa hypothesis, that humans or modern humans originated from Africa and migrated to the rest of the world. Let's look at the classification of humans. Where do you belong, you guys? Where do you belong? Number 1, kingdom, we belong to Animalia. It means that we are animals, okay? Phylum Chordata, it means that we have, chordates means that they have a backbone, that spinal cord. Yes, the backbone. Not basically the spinal cord, but the backbone, because there are some animals which have the spinal cord, but they don't have the backbone. Then we have class, we are mammalia, we are mammals, we have the mammary glands. That's why it's called mammalia. Not boobs. No. In science, we don't call them boobs. We call them mammary glands. Yes. And then order is primates. So all of us, we are under primates, like chimpanzees, the monkeys. Yeah. So family, hominidae. This last part of this word means a family. So the reason why I highlighted this, it means that it is needed. We will ask you this: family to which humans belong. and then Homo, genus. The reason why I underlined or I changed this, it is Homo, because in the phylogenetic tree, we will ask you how many genera are there. How many genera are there? If it's one, it's called genus. If there are many, we call them genera. Then you have which you call a species. We under sapiens. We are Homo sapiens sapiens. So the scientific name is supposed to be written like this. In since you don't have the italics in the paper, so you have to underline it. So we are under Homo sapiens. So the first name is for a genus, and then the second name is for a species. So if they ask you how many species are there, so we will see this when we go to phylogenetic tree. So how do you write the scientific name? This is what I'm trying to say. The first letter must be capital letter, and then for the second name, the second letter must be, the first letter must be a small letter. So this one is a capital letter, while this one is a small letter. Yes, so these colors, they're just there with a reason. Yes, so this one shows the number of years. We start from present, yes, present, and then we go downwards. That's why we say million years ago. M-Y-A, Maya, million years ago. Yes, so then we will see some questions concerning about that, and I will try to explain it nicely as this class ends nicely, so that you can answer any question concerning about that. All right. Characteristics of humans shared with other African apes. Number one, upright posture. We stand upright. That one is very clear. We are going to be a little bit faster. Then we are saying that a large brain. Yes, we have a large brain compared to donkeys, compared to dogs. Yes. So the brain, you see that the elephant has a big head with a big brain. It might not be that the elephant is having a big brain compared to us. No, we are comparing the brain compared to the body size. If our body size is compared to the brain, our brain is bigger. If you compare the brain of the elephant compared to the body size, you'll find out that its brain size is too small. Eyes in front, that is very clear, so that you can see nicely and easily. And then you're saying that eyes with the cones, you can see color. It means that dogs cannot see color. Yes, so but we humans or primates, we can see color. Yes. Then free rotating arms, you can rotate them nicely. Yes, even you can slap someone from behind you. It means that they can rotate. Long upper arms, our arms, the upper arms are longer. Two teeth, yes, we say that we call them teeth. Okay, if you look at the mammary glands on the top, they have two of them. If you look at a cow, it has more than two. Yeah, it has more than two. So it means that the humans and other primates, chimpanzee, gorilla, all those ones, they have two of them, not boobs. Yes. All right. And then they're saying that elbow joints allowing rotation. Yes. This is the elbow we are talking about. Yes, it can allow that rotation. Sexual dimorphism. You can see a difference in a guy and a female, a male and a female. Even if you are a guy and you put things which makes you look like a lady, still we don't know that you are a lady or you are a guy. Even when you talk, even if you twist it, still we will know. So sexual dimorphism, we can see you clearly from a distance. Then you are saying that opposable thumb, you can hold, when you're holding a cup, you see that these fingers this side, the other ones is the other side. So it means that this, the thumb is opposite to other fingers. And then flat nails, though some people, they add some nails there, but you see that they are flat. Yes, we don't have claws. Claws are those for maybe the animals which dig, so we don't have claws. All right. So basically, that's what the similarities between humans and the other primates. What are some of the differences?
Welcome back to our class. So today we are going to be looking at punctuated equilibrium and gradualism. So we are going to compare the two and see where do they differ. Don't forget to subscribe if it is your first time to visit our channel, and don't forget to like our video. So if you look at these two diagrams, one is showing a punctuated equilibrium and then another one is showing gradualism. Gradualism, it shows that a species change gradually. They change gradually over a great period of time. There is no rush in the changing. They change from this point to this point, but the change is gradual. It is not fast. It's just a gradual change. From this point, you see that this rat is trying to change to another color, and then to a lighter color, and then a very light color or bright color. And then if you look at this, still is the same. It shows that there is a gradual change from this point to this point. It didn't happen just in a single event. It happened over a long period of time, over many generations. That is gradualism. While in punctuated equilibrium, you see that the rat started from here. Yes, it's the same as this. So it keeps the same for a long period of time. But this rat is the same which gave rise to this. Yes, so when it reached here, suddenly, yes, within a short period of time, it changed from this color to this color. So when it was here, it changed from this to this immediately without a long period of time. So in punctuated equilibrium, it shows like maybe a mutation occurred, and then the organism changes rapidly within a short period of time, and then becomes like this from this point to this point immediately, and then it remains like that for a long period of time. Or when it reached here, it changed from this color and then went to this color just within a short period of time. While if you look at this, from this color to this color, it took a long period of time for it to change from this point to this point, and also took a long period of time to change from this to this. So if you compare this route and then you compare from here to here, this one shows that it's just a very short period of time. It happened, it changed within a very short period of time, while this one took a long period of time. The same story here. It shows that this rat just changed within a short period of time. It became like this. If you compare it with this, so it took a long period of time. So gradualism, in actual sense, gradualism require a long period of time for it to happen, while punctuated equilibrium requires a very short period of time for it to happen. And then gradualism, it is gradual, while this one, it has some periods of no change, and then periods of rapid change. Periods of no change, if you look at it, and then periods of rapid change. So it has a period of no change, and then has a period of rapid change. So that is what we call gradualism and punctuated equilibrium. Let's look at it. Saying that it explains this punctuated equilibrium, it explains the speed at which evolution takes place. At which speed is evolution taking place? So we are saying that evolution involves long periods of time, where the species does not change or change gradually through natural selection. This is known as equilibrium. So if you look at it, it is from this point to this point. It's the same period of no change. It is just the way it is. Or we can say a period of it's just gradually through natural selection. Gradually, this is the gradually through natural selection. Period of no change, they're talking about this. We are talking from this point to this point, from this point to this point, because these rats are the same. They are the same. They are not changing. But if you look at gradually through natural selection, then this is the period you are talking about, or this is the period you are talking about. So during this period, yes, we call it equilibrium. And then we are saying that this alternates.
it has period of no change. It alternates. This alternates with what you call punctuated. That is short periods of time, short periods of time where rapid changes occur. This is through natural selection, during which new species may form in a short period of what? In a short period of time. So we are seeing that from this point to this point, rapid change happens. Yes, it could be also as a result of what? Natural selection, but species form within a short period of what? Within a short period of time. Basically, that is what we call punctuated equilibrium and gradualism. Gradualism, you find out that organisms, they take a long period of time before they change, while punctuated equilibrium takes a short period of time. There is a long period of no change, and then rapid change, long period of no change or rapid change. They can ask you a question. Explain how did the tiger came up with stripes using gradualism or using punctuated equilibrium. So in this case, for when you explain in terms of punctuated equilibrium, you have to bring the there was a rapid change. You have to include the spots became bigger and then became stripes within a short period of time. So there was just a short period of time, then these tigers with spot became stripes. And then for gradualism, you have to tell us a long period of time. Took a long period of time before these tigers came up with the stripes. Let's look at artificial selection. What is, because we have Welcome back to our class. Don't forget to subscribe if it is your first time to visit our channel.
We are saying that genetics, when we talk about genetics, we are talking about what we call genes. Small sections of DNA, and these sections, they have a certain character they are controlling, is what we call genes. So the study of inheritance, the study of these genes is called genetics. Basically, in this topic, these are the things you need to cover. I'm going to cover the following subtopics in this topic. First one is concepts of inheritance, the terminologies. You need to know the terminologies. Monohybrid crosses, crosses which involve one character, one character. Sex determination, what is the percentage chance for you producing a girl or a boy? How do you determine the sex of a kid? Sex-linked inheritance, the characters which are linked to the sex. Blood grouping, which type of blood group, and then inheritance of blood group. Dihybrid crosses, when we talk about dihybrid crosses, we mean inheritance of two characters. Di means two. Genetic lineages or pedigree. These ones are tracing characters in your family. In most cases, in this case, you're going to be looking at the disorders which can be inherited by the family. So you have mutation. When you talk about mutation, it means change in a part of DNA. It could be the whole chromosome can change, or it can be just a small section of DNA changing. So we're going to look at the mutations. Genetic engineering. Genetic engineering, it means that you're trying to manipulate. So you try to manipulate with the genetics or with the genes of an organism. Then you have a paternity test. Here you're trying to do the testing of the parents, father, mother. If there is any subtopic you don't understand, then it means that you are still lacking, you are susceptible to lose marks, because you don't know which part of genetics they are going to bring. You can bring any part from genetics. So you don't know exactly which part they're going to bring. And if you don't study that part, then it means that these marks which are allocated on genetics, you have lost some marks even before you go to the what? The paper. However, by chance, you can find out that the part you don't know, it has not come. And then you can answer those which you know. But I don't encourage that. So let's look at the introduction. Father of genetics. When we talk about father of genetics, we are talking about Gold Mendel. He did a lot of work on inheritance, and he discovered a law which we call law of inheritance. He concluded genes come in pairs, like when they are, for example, height is being controlled by two parts of genes. When we talk about genes come in pair, a character like, for example, height, size, skin color, they're being controlled by two kinds of genes. So they come in pairs and are inherited as distinct units. It means that one character is not interfering another character during inheritance. Each character is inherited differently, from one from the father and one from the mother. It means that each parent is supposed to give one part of the character. Remember, there are supposed to be two, so it means that the first part comes from the father, the second part comes from the mother. That's what he is saying. So let's look at some of the concepts of inheritance, meaning that the terminologies. So chromatin. When you talk about chromatin, it's a substance within a chromosome containing DNA and a protein. Basically, when you talk about a chromatin, chromatin is DNA. But in this case, this DNA is like threads. Yes, it is in coiled form. When it coils, then it forms what you call a chromatin. Then number two, you have what you call chromosome. Now, when the chromatin condenses, when it shortens, when it becomes thick, it forms what you call chromosome. So chromatin and chromosomes, they are the same thing, but in different versions. So you can say water in ice form and in liquid form. So it's the same thing, but it's in different phases. So we are saying that are thread-like structures of nucleic acids and are found in the nucleus carrying genetic information. So chromosomes are the thread-like, they are like threads crossing each other. And these chromosomes, they carry what called the genetic material. You look like your father because of the genes your father gave you. Then, if you look at this structure, look at this, you might think that this is noodles. Yeah, you think that these are noodles or spaghetti. Yes, but if you condense it, yes, so if you shorten it, you squeeze it, you condense, it forms what you call chromosomes. So when they cross, they look like this. So when you talk about chromosome and the chromatin network, they are the same but in different phases. You see, you can see that what is here is almost what is here. Small ingredients just to make them to be in this shape are being added, just to make them to be chromosomes. But what they carry inside, the most important thing, what do they carry, is the same information as chromatin network. But they form chromosomes during cell division. What about genes? Chromatin network, chromosomes, genes. Genes are just small sections on the chromosome. So if you just break down the chromosome or the DNA, you form different portions. Those different portions, they form what called genes. And then you have, we are saying that this is a small portion of DNA coding or controlling a particular character or a particular protein. So it must be these characteristics you have. You are dark-skinned, you are light-skinned. What causes that? It is genes. And then when we talk about allele, alleles are genes, alleles form chromosomes, alleles form chromatin network. But chromatin network, if you look at chromatin network, you look at chromosomes, you look at genes, you look at alleles, they are the same things. All of them is the same thing, but in a different scale. I'm gonna give you an example. When you talk about that, where do you stay? You say that I stay in South Africa. Where exactly? Then you tell me that I stay in Northwest. Where exactly in Northwest? You tell me that I stay in Rustenburg. Where exactly in Rustenburg? So you keep on reducing the scale. Chromosome, genes, alleles, they are the same but at different scales. So when you talk about allele, it's an alternative form of a gene. You can be tall or short. So an alternative form is called an allele. So we are saying that alternative form of a gene found at the same locus. Locus means the location of a gene, the place of a gene, the position of a gene is what we call a locus. We're gonna bring these things in terminologies, and then please don't forget to give a correct answer. So here is a chromosome. A chromosome is here, but you have different parts, different locations of these chromosomes. So these parts is what you call the alleles. So these alleles may be for height, for size, for color, for blood group. They are supposed to be found on the same position of the chromosome. This side and this side is the same thing. So we call them alleles. So are alternative forms of gene. Then we talk about a dominant allele. So because you have two forms of alleles, one can be dominant, another can be recessive. And both of them can be dominant, or both of them can be recessive. So when we talk about dominant, it means something which suppresses another thing. Yes. So we say that an allele that is expressed in the phenotype when found in heterozygous state. When we talk about heterozygous, it means in different phases. One is like this, one is like this. So it means that this capital letter suppresses this. That's why I say that a dominant allele is that one which suppresses another allele. So this capital letter will suppress the small letter. Then what you call the recessive allele. This one can only express itself in absence of a dominant allele. So if this one is absent, then automatically this small t will also express itself. So we are saying that an allele that is masked in a phenotype when found in a heterozygous state. So it can only show itself in a homozygous. It is only expressed in a homozygous condition or homozygous state. Here is an example. You have homozygous dominant. It means that, you see, they are the same. Homo, I say that when you talk about homo, means the same. Zygous comes from what? Zygote. So it means that this zygote has the same kind of allele. Heterozygous, it means that this zygote has different kinds of alleles. So you have capital letter and small letter. And then homozygous, it's still the same type of alleles, but these alleles are recessive. So it means that it is the same kind. Homo, which means the same. So same, but they are supposed to be recessive. Then when you go to phenotype, when you talk about phenotype, it means the physical appearance of an organism. You are expressing an individual in terms of what you see. In most cases, when you bring a question concerning about a cross, they're going to ask you the phenotype. The first question, the first part you are supposed to write the phenotype. We get these answers from the question. Is it clear? So we are saying that physical appearance of an organism is determined by the genotype. For example, tall or short. You can be tall, you can see that a person is tall. You can be short, you can see that a person is short. You can be light skinned, a person is light skinned, you can see them. And then what about genotype? Here you are describing an organism in terms of what? In terms of genes, genotype. The gene type. Which kind of gene type do you have? So here is genetic composition of an organism. So you know you are tall, but which kind of tallness? You might be tall, but homozygous tall or heterozygous tall. You have an allele for shortness. You can be short, but you have an allele for tallness. You see two people who are short, they produce a tall kid. Two people who are dark skinned, they produce a light skinned kid. Two people who are light skinned, they produce a dark skinned kid. And then the guy can say that, this is not my kid. It's whose kid? All right. So here is another example. We'll talk about this. Describing this in terms of the genes is what called the genotype. Then also this is the genotype, and also this is what? Genotype. So I will say that this is homozygous dominant, this is heterozygous, and then this is homozygous recessive. So I'm describing these flowers in terms of genetic composition. So then when we talk about homozygous, I've been talking about this. Homo, which means the same zygous from the zygote, then it means that this zygote has the same kind of genes. Two alleles, two identical alleles for a particular characteristic. Homo, same, can be homozygous dominant or homozygous recessive. When we talk about hetero, when you say hetero, it means different. So the zygote has different kinds of alleles. For example, capital letter B and then small letter b. So they are different letters or different alleles. If you look at this picture, you'll find out that homozygous, you have this. They are found on the same locus, same location. This one and then this one, and then this one and this, you see. While if you look at heterozygous, same location but different letters. Same location but different letters. So this is homozygous recessive, this is homozygous dominant, and then this is heterozygous. For the case of heterozygous, we don't have recessive and dominant, we just say heterozygous. A monohybrid cross is a cross which involves one character. You are right, it involves one character. But now you said pink and white. It still is fine. The answer was correct. But where is the character there? The character is color. That's the character. But this character has two alternative forms. The blue you talked about and the white you talked about. It's either blue or it's white. So those are the what called the alleles. So the character is the color of that individual you talked about. And then alleles are the different types of colors you have talked about. You understand? So you can visit our website www.sandaeduc.com, and then if you have any challenge question, then you can also WhatsApp on that number that is MSID. So I'll be able to try to answer your questions. Let's look at meiosis. Meiosis, we said that meiosis, humans reproduce, or other organisms reproduce. They multiply. What about cells? Cells, they have two kinds of reproduction they undergo. They have what called mitosis and you have what called meiosis. Mitosis can occur in other body cells, but meiosis occurs only in the reproductive cells. Where did you come from? Actually, meiosis was the basis of you living. Without meiosis, you wouldn't have existed. So let's look at meiosis in detail. So it's a type of cell division that results in 4 daughter cells, each will have number of chromosomes to that of the parent. So it means that meiosis starts with the one cell. This cell divides and then you form 4 daughter cells. These cells, they have the chromosome number to that of a parent. If you have started with the 4 chromosomes, it means that these cells, they will result in 2 chromosomes. That's why you're saying that half the chromosome number of that of the parent. If the man, the body cell is having 46 chromosomes, then the reproductive cells or the daughter cells which are resulting from that are going to be having 23. And then from the mother also 46, they are going to result into 23. So the process of reducing these cells into half the chromosome number, that process or that reproductive process is what we call meiosis. So what does meiosis result in? So this meiosis will result in the production of gametes. Gametes, these are the sperms and the ova. And when you're talking about the humans, and then plants, you're talking about spores, the pollen grains. You can talk about the ovules. Those are some of the things which are as a result of meiosis. So in humans or in animals, it occurs in the ovary. See this colored word, in the ovary. Basically, ovary are found in female, and then these ovary, they produce what you call the ova. So the reproductive cells in the females, we call them ova. If it's one, it's called the ova. If there are many, we call them ova. And then in male, we call it's occurring in testes. And then in testes, what we call them spermatozoa. The cells which are being produced, we call them spermatozoa. So in female, we call them ovary. Those are the ovaries. The ovaries produce the ova, and then in the testes, they produce what we call the spermatozoa. What about in plants? In plants, occurs in the ovary still, but what it produces, ovary produce what we call the ovules. Meiosis occurs in the anthers, and then what it is being produced is what you call the pollen grain. So the question can come and say that other than the ovary in animals, which other place does meiosis occur? That is testes. And what does it produce? It produces what you call the spermatozoa. And then in plants, it occurs in the ovary. It produces what you call the ovules. Basically, that's what you needed to know about this first introduction of meiosis. You know, meiosis is basically the cell divides, and the most important thing to divide is the nucleus. So we need to know the nucleus to do a recall about the nucleus, because we saw the nucleus at the beginning of DNA and also saw the nucleus in the grade 10. So let's look at the nucleus, the centrosome, and then the cytoplasm. Nucleus contains the genetic material. What about cyto means a cell, and then plasm means a fluid, the fluid of the cell. So basically, this is what you are going to look at. Let's look at them in detail. So this is a cell. It has a nucleus. Outside, you have what called a cytoplasm. Then in the cytoplasm, you have what called the centrosome. Centrosome, why do we talk about the nucleus, centrosome, and the cytoplasm? Because the DNA, you'll find it there, and then it is the centrosome which produces the centrioles, and the centrioles produce spindle fibers which hold the chromosomes in position. That's why you talk about it. And then there is a time when this one disappears, and then you remain the content of the cytoplasm. So the nucleus disappears, and then you remain only with the content of the cytoplasm. So basically, that is the nucleus. And then when it brings it in exam, it looks like this. So this is the nucleus. It's the nucleus, but the nucleus has a dense body which we call the nucleolus. Then outside the nucleus, you have the nuclear membrane, which has the extension which we call the endoplasmic reticulum. But because it has the ribosome, they look rough. Then we call them the rough endoplasmic reticulum. The rough endoplasmic reticulum. So basically, that's it. It has the nuclear pore, the holes where substances go through, most especially messenger RNA in this case, and other substances. Then you have what you call the nucleoplasm, the plasma of the nucleus, meaning that the fluid of the nucleus. The nuclear envelope, that's what you call the nuclear membrane. Then the ribosome, we say that the ribosome, the sites for protein synthesis. Chromatin, this one is the one which condenses to form chromosomes. And then you have the nucleolus. Then you have the rough endoplasmic reticulum. So we have explained all that. So when you look at the nucleus, or when you look at the DNA, basically that's what you see. You have, when the chromatin network condenses, you form what you call the chromosomes. So the chromosomes, they condense. You see, they have condensed, and then after that, you form the real structure of a chromosome. It looks like this. So that's the structure of a chromosome. Now let's look at the chromosome in detail. So we have seen where the chromosome come from. It cannot exist without DNA with its specific genes. You see, it has specific genes. So let's look at the structure of a chromosome. Why do we need to look at it? Chromosome? You resemble your father because of the chromosome, because of the genes you obtained from your parents. Your father, your mother. What about those who don't resemble the father and the mother, but they resemble their grannies? We shall see. Another question. What about why? So in this topic, we're going to see why. Why do you resemble your neighbor? Why do you resemble when you go somewhere? When you go somewhere, someone can say that, hey, when you resemble someone. Why do you resemble someone? Those are some of the questions we are going to answer. And this is the reason why we need the structure of the chromosome. So the structure of the chromosome. Chromosomes are thread-like structures present in the nucleus. They're thread-like. They're thread-like. It means that they are like strings, thread-like structures found in the nucleus. That's why we talked about the nucleus, which carry genetic material. That's why you resemble your father. That's why you resemble your mother, which carry genetic material from one generation to the next generation. So it means that your kids will try to resemble you. Yes. Then you're saying that each chromosome is made up of the DNA. We saw this, that DNA genes are almost the same thing, but on a different scale. So these DNA or these genes are tightly coiled many times around a protein called histones. So the histones, we saw this when we are talking about protein synthesis, and also when you're talking about the structure of the DNA. Yes, so they coil around to form what called the histones that support the structure of the DNA. Because the genes cannot just exist there in air, they need to have a support. So the support we call them histones. So these are the support which make the DNA strand to stand or to be where it is, and so that the chromosome can be formed when this DNA condense. All right, this is the structure of chromosome. Before, remember, we talked about during DNA replication that when does it occur? We say that it occurs before the cell divides or before meiosis. So before the cell divide, the chromosome will look like this. Yes, they will look like this. Therefore, we call this one single-stranded chromosome. Chromosome 1, chromosome 2. Sometimes we call them unreplicated chromosomes. They have not yet done DNA replication. But the moment DNA replication occurs, this chromosome is going to change from this format to this format. That's why we say that it doubles the chromosome number. Don't say that it increases the chromosome number. It increases the chromosome number. It increases the genetic material. From what to what? The best answer is double the chromosome number, doubles the chromosome number, so that when it was like this, then it becomes like that. So this is the centromere, the center of the chromosome, we call it centromere. The two chromosomes which look alike, it means that they share the same structure, yes, and the position of the genes on them, then we call them homologous chromosomes. Homologous chromosomes. So one chromatid from one chromosome and another chromatid from another chromosome, because they are homologous chromosomes. Therefore, because we are talking about chromatids of homologous chromosomes, then they are going to become homologous chromatids. Why? Because we say that they are coming from homologous chromosomes, but each part is a chromatid. Therefore, they become homologous chromatids. So so sometimes, as I say, that this is called unreplicated chromosomes, and then we call these ones the replicated chromosomes, or what you call homologous chromosomes. So each of these has two names: unreplicated chromosomes or single-stranded chromosomes, and then these ones double-stranded chromosomes or replicated chromosomes. So the number of chromosomes in each cell is a characteristic of organisms. For example, humans, we have 46 chromosomes. You'll find out that Drosophila melanogaster has 8 chromosomes. You'll find out that the sheep has over 60 chromosomes. So each organism has a specific number of chromosomes it has. And you might find out that the biggest organism has fewer number of chromosomes. So that's how life, or that's how God is. So it doesn't matter whether you are big or small, but you might have your own uniqueness. We are saying that chromosomes which are single thread or strand become double when the chromatid joined by centromere. So the center of the chromosome, we call it centromere, as a result of DNA replication. So if DNA replication occurs, as we said, now the single-stranded chromosome are going to become double-stranded. So that's why you call it unreplicated chromosome or single-stranded chromosome. The moment DNA replication occurs, it becomes double-stranded chromosome or replicated chromosomes. Yeah. So what are some of the difference between the haploid and diploid? Di means two, ploide means you are describing a body. Yeah. So half means it is derived from the word half the chromosome number. If a human being is supposed to have 46 chromosomes, so when you are haploid, or when the structure is haploid, it has the half the chromosome number of that organism. For example, if an organism has 20, the haploid state of it is going to be 10. If the organism is having 32, the haploid number of that organism, or haploid state of that organism is going to be 16. So haploid means half the chromosome number, diploid means doubling. Doubling. What does it mean? Where is the double there? It means that half from the father, half from the mother, and then they double it. When they double, then they form what called the diploid or diploid, the way you pronounce it. So haploid is the quantity of the cell or organism having a single set of chromosomes. They have what called a single set of chromosomes. So if you have chromosome number from 1 to 2, from chromosome number from 1 to 10, these chromosome numbers, these chromosomes must be in pairs. So if you are haploid, chromosome number 1 to 10, they will be in single. They will be 1, 1, 1, 1. They won't be in pairs. So that's what you call a haploid. But if it is diploid, is when the cell or organism has a paired or two sets of chromosomes, one from each parent. It means that one from the father, one from the mother. When they combine, because there are two organisms contributing this, then you form the word di, which is two. And then ploid means a body. Yes, it's a state which you describes organism with two sets of chromosomes, one from the father and one from the mother. That's the meaning. So for sexual reproduction to take place, sexual, it means that it must include the gametes. It must include the gametes and must include the sex. Reproduction to take place, yes, what happens? A haploid male gamete, that is, if we are talking about humans, we're talking about sperms, will fuse with a haploid. It means that it will refuse. When you say a haploid male gamete, half of the chromosome number from the male, the gamete will have half of the chromosome number from the male. If the male is 46, the gamete is going to have, or the sperm is going to have 23 chromosomes. Will fuse with the haploid number from the female. Also now the female is going to produce 23. When they fuse together, you form fertilization. So when they fuse together, that process is called fertilization. Now you form what you call a diploid, because half from the father, half from the mother. Then because it's half from different people, then it becomes di, becomes two. They fuse together. And then when we're talking about fusion or fertilization, we are not talking about sex, because the fusion or fertilization can take place without sex. Meaning that maybe they used a male just give out the sperms. So when you're talking about fertilization, we're talking about sex has already occurred, and then the sperms, they swim along the female reproductive system,
and then fusion will take place. This fusion can even take place. Someone had sex at night, and then you're walking on the street, fusion is taking place. Imagine, did you know when was your fertilization took place? You don't know. Even your parent doesn't know. Okay, those are the wonders of the science. All right. If you need more questions concerning about this reproduction, go and visit our YouTube channel, which you call Bright Doctor. Bright Doctor. I'll give you the link in the description below so that you can understand it better. All right. The result is a diploid zygote. The result is a diploid zygote. So when you have a haploid, you form a diploid or a diploid zygote. So it means that when the sperm fuses with the ovum, you form what you call a zygote. But this zygote has a diploid number of chromosomes. This body has a diploid number of chromosomes. It means half from the father, half from the mother. All right, haploid, half, which is indicated, haploid is indicated as 1N, and then diploid is also indicated as haploid from the mother, 1N. For example, haploid from the father, for example, is also 1N. So when you combine them, you form the diploid, which is whole. Yes, it's the whole. Now half and half, you get whole. Yes, basically half from the father, half from the mother, from the diploid. Di means two, meaning that contribution from one person and then contribution from another person. So because this resultant body is having two contributions, then we call it a diploid. So it is indicated as 2N. We shall be using this language. So you have to know when someone uses this language, you must know what it means. So a sperm, ovum, and then you form a zygote. Zygote is where a fetus develops from. We shall see in the videos we're gonna be releasing soon. How is the zygote form? You see the sperm swimming after sexual intercourse. You see the sperm swimming going to the ovum, and the fertilization takes place. For more detailed about this, visit what you call the Bright Doctor. Bright Doctor, just a link below. Drop your question, then we shall give you the answers on that YouTube channel. But for purpose of passing exams, we don't go far away from this. But there we can describe as much detailed as you want. So sexual cells is either sperms and ova are haploid. So we call them haploid. They are 1N, 23 chromosomes in humans. So it means that the other organisms have different chromosomes. So we have what called the somatic cells. So it means that we have two types of cells. We have what called the sex cells. Those are the gametes. I say that meiosis can only take place in these cells. We don't find meiosis in any other cells, only in the gametes. So you have the other body cells. Soma comes from the word soma, which means a body. Yes. So these are body cells are diploid, which means that they are 2N. If they are 46, it means that the haploid is going to be 23. But the question when it comes, it does not bring only human. You can only talk about 46 if they have specified in the question that this is a human cell, because they can bring a cell with 10 chromosomes. They can bring a cell with 4 chromosomes. If they don't specify that this is a human, please don't use 46, because the question is not asking about humans. Then you have the 23 pair of chromosomes that result in the zygote are divided as follows. How? Number 22 pairs. These are the number of chromosomes we have in the body. 22 pairs. That is from pair number 1 to pair number 22. We call them autosomes. Autosomes. It means that how many chromosomes do we have? 44. Because this is a pair. Pair number 1 up to pair number 22. We call these ones autosomes. One pair of sex chromosome, that one pair, that is the 23rd pair, we call it sex chromosome, or scientifically we call them gonosomes. Scientifically we call it gonosomes. So this pair is divided into two. We have what called the XX, which is female, and then you have XY, which is male. So it means that females always, they will have XX, and males will always have XY. There is a question which is supposed to be posed here. Why, if females are XX and the males are XY, it means that it's the males to determine the sex, because female have X, also male has X, but female don't have Y, but male have Y. So it's the males to determine the sex of the human being, the sex of an individual. So males, you must stop saying that ladies are only giving you females, they're only giving you boys, they're only giving you girls. Say, I'm giving myself boys, I'm giving myself girls. It's you who is determining this. However, there are some other factors which also determine. It can be on the side of the female. For more details, visit the Bright Doctor. Drop your question there. We will be able to answer those questions. If you drop the question there, then I'll be able to answer those questions. Humans have what we call the chromosomes. We say that from chromosome number 1 to chromosome number 22. These chromosomes are arranged in a specific order, and they appear in a specific way. They are in pairs, and then they describe different characteristics in the organism. So let's look at the karyotype, the composition of the nucleus. Which kind of nucleus do you have? The chromosomes, the type. Karyo is derived from the nucleus, and the type. So which type of the nucleus do humans have? That's derive the name of karyotype. Humans cell are made up of 46 chromosomes. It means that our nucleus have 46 chromosomes, and then these 46 chromosomes are arranged in pair. These chromosomes are arranged in pairs. It means that they cannot exist as singles. What does it mean if you're not married? All right. It means that they are arranged in pairs. Each pair is different from each other. Each pair is different from other. So these pairs are different. How do they differ? How do they differ? They differ by shape, size, and genetic composition. So pair number 1 is totally different from pair number 2 by size, shape, and the genetic composition. It means that what pair number 1 determines is not the same thing what pair number 2 determines. For example, you'll find out that chromosome number 23, pair number 23, will determine the sex. You cannot find this in other chromosomes. So they vary in shape, size, and the genetic composition. From pair number 1 to pair number 22, number 1 to 22 are called autosomes. What about 23rd pair is called gonosomes, which has XX for female and the XY for male. X, you have to know this, X is longer and bigger over Y. Why? X is longer and bigger over Y. Question, why? Why is X longer and bigger? Bright Doctor will answer that. But on the purpose of this, X carries more genes compared to Y. So a karyotype, an example looks like this. You see that chromosome number, pair number 1, number 2, number 3, number 4, they vary in appearance, shape, and also they vary in the genetic composition. So from 1 up to 22, we call them we call them gonosomes. We call them autosomes. And then the 22nd pair, we call it gonosomes. So basically, that's it. Revision of the process of mitosis. It's advisable before you start meiosis to do a revision concerning about the process of mitosis. So guys, our next video, as we say that we're gonna be having prizes, airtime, meals. Yes, we send you a voucher and then you buy whatever you want to buy. It can be a school bag, t-shirts. We have a lot of gifts to give out on our YouTube channel. So please don't forget to subscribe. Don't forget to subscribe, like, share, so that others also benefit from this content. And then we're gonna be having all the subjects. So we are trying to increase this. So mitosis occurs in every living organisms. In the somatic body cell to produce, those are the body cells, other cells other than sperm and ovum, to produce genetically identical cells. So you can have a hole on the mouth, on the cheek, and then you need meat. So what they can do, they can cut the bum and then they put on the mouth. So you move with the bum on your face. Basically, why do they do that? Because these cells, they undergo what we call mitosis. They are genetically identical. The meat from your bums is exactly the same as the meat from your face in terms of the genetic composition. So two identical cells are reproduced. Two identical cells are produced with identical chromosome number equal to the original cell. It means that the parent and the babies or the daughter cells, they're exactly the same. There is no difference. That's why when you get a wound, another meat develops. Those cells which develop, they're exactly the same. Mitosis has only one cell cycle, i.e., it doesn't have that mitosis 1 and mitosis 2. No, it only has one cell cycle. Only one cell cycle. It means that meiosis, when on the side of meiosis, it has two cell cycles, meaning that it has meiosis 1 and also has meiosis 2. So guys, let's look at the process of meiosis. Yeah. The process of meiosis was a very good introduction of meiosis. Now let's look at meiosis in detail. And this will be our next topic tomorrow. Don't miss it. Don't forget to subscribe, like, and then share the video. We will be having gifts, prizes in our next videos, starting from
Welcome back to our class. Don't forget to subscribe if it is your first time to visit our channel. Let's look at artificial selection. What is, because we have talked about natural selection, nature selects which one is best suited to the environment. But here, artificial selection, it's the human which selects which one is supposed to continue with life or not. Which characters do you need? Actually, it depends on the desire of the human being. It's where humans select for a desirable characteristic or select for desirable traits in agricultural products, animals, rather than leaving the species to evolve naturally or gradually without human interference. So in this case, we have humans interfering what is happening in the environment. So humans select, maybe I want birds with or chicken with big eggs, which can produce big eggs. So I'll be selecting them. For a long time, humans have been using this kind of method during breeding experiment to develop organisms with a selected set of desirable characteristics. So they keep on selecting those which are having desirable characteristics, then they select them and then they keep them. And those which don't have desirable characteristics, they remove them from the system. An example, you can find that they do select because of the following reasons. Number one, increase the quality and the quantity of milk produced by cows. Sometimes you can ask you the functions of artificial selection, the benefits of artificial selection. So these are the things you can give us that results in increase the quality and the quantity of milk production or produced by the cows. Then also drought-resistant organisms, they select for those organisms which can resist the droughts. And then disease-resistant organisms also select, they want organisms which can resist diseases. Yes, so these are benefits also of artificial selection. And then increase sugar content in the sugarcane, because they want to produce a small sugarcane can produce a large amount of sugar. So these are some of the examples. They started with these dogs. You find out that they want these characteristics. So these dogs which have such characteristics, they are being kept, they are being bred, so that after so many generations, you end up having such. The same story here, they started with this. You see 1957, the chicken was like this. 1978, it was like this. 2005, it's like this. So it means that the one which can have big meat or can produce meat, then they are being what? Selected for. And then those ones which can produce big meat or large meat or high quality meat, then they remove them from the system. The same story here to the maize and the wheat. So this is the process. What happens? Suppose you wanted a variety of cow that can produce a lot of milk. So what happens? You choose or select the cow in your herd that produce most milk. So you are looking for the cow which has the characteristic you are looking for, or a character you're looking for, and then you let these cows reproduce. You let them mate, and then they reproduce. Then they will produce kids. They select select the offsprings that produce the most milk. So the offspring will be produced, and these offsprings will be having those which can produce more milk and those which cannot produce more milk. So those which can produce more milk, you keep them. So you keep on selecting them until the genes become pure. Then you're saying that you let only these offsprings to reproduce. The one which can produce milk, you let them reproduce. Keep repeating the process of selection and breeding until you achieve your goal, until those animals which can produce milk are obtained. So what are some of the differences between natural selection and artificial selection? So we have seen that in natural selection, environment, yes, it occurs in environment. So the environment or nature is the selective force, while in artificial selection, humans select. So human is the selective force. Here in the environment select which one is the best suit, while this one is human. Like desirable characteristics. And then number two, selection is in response to what suits to the environment. Which character can survive in the environment, while here it satisfies the human needs. It's not about which character is suitable in environment, no, what sustains the human needs. And then this one occurs within the species, while this one's it can occur in one species, more than one. Yes, because you can crossbreed from between two species. Yes, but while this one occurs in one species, strictly in one species. Yes, so that's some of the differences between artificial selection and natural selection. Let's look at a speciation.
Out of Africa hypothesis and the evidence for African origin of modern humans. Modern humans originated from Africa and migrated to the rest of the world. That is out of Africa hypothesis. This states that modern human, guys, you need to put the word modern. If you just say humans, then you are wrong. You have to tell us modern humans, wise humans. Modern humans originated from Africa and migrated to the rest of the world. That is the out of Africa hypothesis. That's how it states. What shows that they originated from Africa to the rest of the world? Those are the evidences. Let's look at the evidences. Okay, evidences to support this hypothesis. We are saying that number one, fossil evidence, genetic evidence, and then cultural evidence. We have talked about this. So it's just repeating and repeating. Guys, don't you see that these guys, they're just making you just to pass this paper? There is no way how you can fail. Have these slides, read them, then go to the paper, and then we see. Practice questions, yes, and then we see. Number one, fossil evidence. What can you say about that? Fossil of Adipithecus were only found in Africa. Remember, Adipithecus is the first fossil we studied about. That is the oldest fossil, yes. Fossil of Australopithecus, for example, Karabo, Little Foot, Tongue Child, Mrs. Place, all these Remember, they didn't say that Australopithecus africanus, Australopithecus. Carabo is Australopithecus sediba. Littlefoot, Africanus. Africanus, and then this one also is Africanus. So Australopithecus were found in Africa. And then Homo habilis were only found in also Africa. So all these oldest fossils were found in Africa. It shows that modern humans originated from Africa and then they went to the rest of the world. The oldest fossil of Homo erectus, although they found some Homo erectus somewhere else, but the oldest was discovered in Africa. So it means that Homo erectus was also in Africa. It came from Africa. Because it's Homo erectus, it can walk on two limbs, then it migrated to the rest of the what? Of the world. It can walk faster, so it migrated to the rest of the world. Homo sapiens have been found in Africa, still oldest in Africa, which shows that, oh, yeah, we originated from Africa and migrated to the rest of the world. All right, genetic evidence. Let's talk about genetic evidence. We saw about this. Mitochondrial DNA, that is abbreviation like this. Then you're saying that is inherited only from the maternal line. We saw this. And then analysis of mutations of this mitochondrial DNA. Yes, if you analyze the mitochondrial DNA, yes, what happens? It shows that the oldest female ancestor located in Africa. We said that Eve originated from Africa. And you're saying that all humans descended from her. Who is that? The Eve. Or the Eve which we know. All of us, we know Eve. Don't confuse this Eve with the Eve, the bird. Birds are under class Eve. We're not talking about birds. We're talking about Eve. Adam and Eve. So then you're saying that cultural evidence, we saw it also. Using tools with cutting and sawing improves human nutrition. Development of tools, stone tools became more sophisticated as hominins evolved. So as the hominins tried to evolve, then even these tools were becoming more advanced. These tools require planning and thought, which means that they require a big brain. That included sharpened stones placed on wood. Yes, they make a wood and then they sharpen the stone, then they put it on the wood and then they make a spear. Yes, and remember, if you watch those, you like adventure movies, you can find those kind of spears. Yes, and then some of them, they use them as axe. Axe is the one. Some of you don't know axe, but axe is used to cut trees. From this point, the tool progressed to knives and blades. Now they can now use it to remove the skin, to kill animals. This was being used by Neanderthals. If you watch that video, you'll be able to see this in Neanderthal we are talking about. Used for hunting and protection. Yes, so they are being used as protection measure. Phylogenetic tree, guys. Phylogenetic tree, guys. I really need more time on this. What is a phylogenetic tree? It's a schematic form that shows the evolutionary relationship within a set of organisms, groups of organisms. It could be a set of organisms or groups of organisms. So it shows where you really came from until where you are now. Note, you just need to understand how to apply this phylogenetic tree. You don't need to memorize what is on the phylogenetic tree. No, you just need to how it operates. That's it. You don't need to cram anything. Okay, let's try to interpret phylogenetic tree. Sample number 1. This is, we count from 0. We don't count from this side. And then we count from this side going downwards. Yes. But this tree starts from this side going this side. So we are saying that Australopithecus afarensis originated between 5 around, so I can't say 5.5 because 5.5 is this side. So this is 4 4.5 and then 5, 5.5 then 6, because 0 is this side. So we are saying that maybe around 4.8 here, originally around 4.8, yes. And then this Australopithecus afarensis gave to Homo habilis and also gave to chimps, yes. The Homo habilis gave to Australopithecus a robustus. All right, Homo habilis gave rise to Homo erectus, the upright walking man, and then Australopithecus robustus, and then Homo erectus gave rise to Homo sapiens. So now we can say that's how we can interpret it. But we have a lot of questions, here. Which one is the common ancestor of all these species? I will say it is Australopithecus afarensis. And ask. Okay, let's go to another phylogenetic tree and then try to interpret it. Here, 0 is this side now and is on top. And then 1, 2, 3, 4. Yes, so these are million years ago. And then it means that in this case, they can ask you how many genera are here. 1, Australopithecus 1, Homo 2, Kenya, Theropus 3, and then it's Australopithecus. All these are Australopithecus 3. This is Homo. So it's 1, 2, and then 3. So there are 3. All of these ones, there are 3 genera. 3 genera. Genus is 1. The first name, as long as it is repeating, then it means that you have counted that one. How many species? Species a lot. All of them, they are species. So you can count them. That Sapiens 1, Neanderthalensis is 2, Augusta 4, Habilis 5. So you can count them. You can count them. So the second name, Robustus. So basically that's it. And then you can ask you another question. Which species competed with the resources with Homo sapiens? You draw a line. You find out that Homo Neanderthals, it was there. You find this one was there. If you draw a line, I think even this one touches. So it competed the resources with that. If you look at this one, which one competed the resources with Homo habilis? I draw a line touching here and I draw a line touching here and then on top. So I can count. So how many million years did Homo habilis exist? So I count from here up to here. From here, I draw a line to this, and then I draw a line to this, and then I find a difference. From here to here, and then from here to here. So from here, it may be 2.5, and then here, 1.5. So I will say 2.5 minus 1.5. So it's gonna be 1 million years. So how many million years ago did the Homo habilis extinct? So I'll say that Homo habilis extinct because I'm going this side. Extinction, I'm going this side. So I'll draw extinct. It started from here, it's extinct from here. So I draw a line going up, maybe it is 1.5. So I'll say 1.5 million years ago. If they are talking about extinction, you have to tell us million years ago. Time of living, million years. Million years. If it is extinction or existed, when did it exist? So tell us million years ago. When did it exist? I draw a line touching this 2.5 million years ago. How long did it exist? I find where it started and where it ended. I find the difference. 2.5 minus 1.5 gives me 1 million years. So basically that's it. And then if you look at this one, is also another phylogenetic tree. Okay, I'll come back with that. All right, another phylogenetic tree. In this one, we start from here on top going downwards. You see, we have different phylogenetic tree. So guys, it should not confuse you. It should not confuse you. So it means that this is the Sahelanthropus tacadensis is the origin. So no, it's not the origin. Origin is here. It gave rise to this and then this. So it gave rise to this, and then this one gave rise to this, and also this. So this, how many genera are here? I'll find out. 1, 2, 3. Adipithecus, 3 is still 3. Australopithecus, 4. Paranthropus, 5, and then Homo, 6. There are 6 genera. Which organisms existed at the same time as Paranthropus boisei? So I draw a line because now this scale is this side. I draw a line touching the scale. I draw a line touching the scale. So I find out that is this. and then even here, this side. So whatever cover is covered, then it means that even if it's just a part which is covered, then it means that that organism is also involved. Then lastly, this one is trying to show you what happened to the brain, what happened to the tools, and what happened to the skulls. So it's just a combination of everything. So now they're saying that you have Adipithecus remedus here. It means that you still have some other there. Here we don't know, it means that there is a missing link there. And then yes, Australopithecus anamensis. So it gave rise, this is the period it existed. So the bar shows the period it existed. Which one existed the longest period? You'll find out that Paranthropus boise. Yeah, they existed for the longest time. Existed for the longest time. Homo sapiens, Homo erectus, and Paranthropus boise. Which ancestor is for Homo erectus and Paranthropus boise. So I'll come back here like this, I go back. As I also come like this, here is an ancestor, but do we know it? We don't know it, the question mark. I continue, here is another ancestor, we don't know it. Then until we know, we reach where we know. It is Australopithecus afarensis. So it's the ancestor, the common ancestor of Homo erectus and the Paranthropus boise. What about Homo ergaster? Homo ergaster and the Paranthropus robustus. Homo ergaster is here, so still I will go back in the same way. So you see it goes back to the same level. Oh, what about Homo erectus and the Homo neanderthalensis? What about them? I go back like this. I have to continue until where I reach Homo ergaster. So Homo ergaster is the common ancestor for Homo erectus and the Neanderthals. All right. Now let's compare this with this. Which one first used tools? Which one first used? We know that Homo habilis first used tools, but according to this, what are they saying? These are the oldest tools. Older ones are the oldest tools, and then you have these ones, and then now you have the biggest ones. The technology is here. So they're saying that if you look at which one, this one started to use tools from here. So I can draw a line according to this, yeah. Paranthropus, this one, and then this one also you used tools, yes. And also Homo habilis, you see, also Homo habilis also use the tools. So these ones, the one you draw a line touching the what? The scale. So that's how you can interpret that. Then they can ask you which ancestors started to use two limbs by bipedalism. So you go draw a line is here, started from here, then draw a line. It means that Australopithecus anamensis started to use two limbs. Yeah, if you watch that video, it will show you exactly. And then just before that, they were using four limbs, quadrupedal. And then you have, so that's how you can interpret. Draw a line, and then you interpret. And then here are some of the questions. Let's look at the questions. Then you're saying that, let's look at the question. What is the name of the diagram? Is phylogenetic tree. How many genera are shown in the diagram? You count the genera. Adipithecus, that's one. Adipithecus 1, Australopithecus 2. If you check it here, Adipithecus 1, Australopithecus 2, Homo 3, Paranthropus 4. So there are 4 genera. That's basically that. They're saying that what is the common ancestor for Homo sapiens and Homo erectus? I think now you know how to find it. Homo erectus here and the Homo sapiens are here. I draw a line until they touch. Homo ergaster is the common ancestor. What is the common ancestor for all species? All species. So Adipithecus remedus. And then give 2 pairs of species that competed for resources. Just choose. If I choose this, I draw a line. Yes, it competed. This one, boise, competed with robustus. Still competed with Homo habilis. Still competed with Homo ergaster. You see, so you just draw a line, those 2, then you find out that is the answer. Which species first use tools? Which species first use tools? In actual sense, it's supposed to be Homo habilis if they are talking about homos. But if they're not talking about homos, Then if they are saying tools, you see these are the tools, old ones. Then you draw a line, all these ones. You see which one first used. I think the one which first used tools is this Paranthropus. Yes, Paranthropus. Yes, it's the one which first used tools. I think even this one, yeah. Australopithecus africanus is the one which first used tools, and this also, two of them at the same time. And then they're saying which species first walked on two limbs. Then you go to the two limbs. Bipedal is here. You see, bipedal started from here. So it's Australopithecus and Amensis. Then they're saying that how many million years, Maya means million years ago, did Australopithecus africanus appeared? Australopithecus africanus. I look for Australopithecus africanus. It's here. So I start from the dark line. Yeah, so they are saying that maybe it is 2.9. Yes, it's not 3. It's not yet 3. It's 2 because I count from this side. 2.9. Yes. Then they are saying that 2.9 million years ago, 2.9 Maya. When did the Homo habilis became extinct? When did the Homo habilis became extinct? Where is Homo habilis? You look for it. Homo habilis is around here. Yeah, it got extinct. You draw a line. Maybe it's 1 point because I count from this side, from top. You count from 0. It's 1 point, 1.5. You draw a line. You must use a ruler. Don't just estimate like me. You must use a ruler. Then they are saying that how many million years did Australopithecus africanus exist? Australopithecus africanus. I go Australopithecus africanus. I draw a line on head. I draw a line on bottom. The bottom is 30, and then the head is around 2.5. So 3 minus 2.5 is 0.5 million years. How did the brain size show evolution trend? So now they are not talking about here. They are saying that brain size, you see that expansion increases. So how did it show evolutionary trend? Increase in the evolution. So you just have to answer from your theory that increase in the brain size indicates that more brain cells are formed, or it has more brain cells, which indicates a high intelligence. That's it. And then they're saying, give one example of transitional fossil. Still, this one is from your theory, Sediba, or Australopithecus sedibus. Also, A, Sediba. Capital A, full stop, Sediba. That's the meaning. Or Carabo. If they ask you scientific name, because sometimes they ask you scientific name, sometimes they ask you just the what is it. Then if they ask you what is it, without specifying scientific name or common name, just give the name. Or you can give the scientific name, whatever. What is easier for you? How did the dentition show the diary of the species? The dentition, the kind of teeth. If you look at the other primates, they had these canines, tough jaws. It shows, it means that they were feeding on rough, tough food. Yes, so that the canine can bite. Yes, while Homo erectus. Homo sapiens, the dentition, the small canines, even the shape of the jaws, it shows that they are feeding on cooked food or soft food, cooked food. While the other ones, the one like, if you have well-developed canine, then it means that you are feeding on a tough, rough. It's a raw and tough food. So the dentition will show you which kind of food you are feeding on. All right, guys, let's look at some of these questions now. I'm going to open the what? Now it's time for discussion. It's time for discussion. Someone saying, sir, don't mind. All right, guys, it's time for discussion. If you know that where you are, they are making noise, you let me know. Here's the questions. I'm gonna get someone and then try to answer like 3 questions. Everywhere. I'm saying the table below shows.
Yes, sir.
Let me try to interpret for you, and then I ask you questions, you answer. They're saying that don't give me answers from that booklet, from that slide down. No. I want your own answer. Table below shows the evolutionary evolution of cranial capacity in some species. You have Sahelanthropus, you have Australopithecus, Homo habilis, Homo erectus, Neanderthals, and then Sapiens. All right, they're saying the period of existence. This is the period of existence. They existed 7 to 6 million years ago. So, and then this is the brain capacity. So you see that the brain capacity is increasing. And remember what happens if the brain capacity is increasing. You know what we try to discuss about. So they're saying, give two hominid genera, we say that is originating from what?
These cells are genetically different from each other and from the parent. The process of meiosis, we say that a type of cell division that results in 4 daughter cells, each cell with half the chromosome number to the parent. So meiosis is a continuous process. It's a continuous process, but it's divided into different phases and events. So the events, for example, the first event is what we call the interphase. Interphase is not part of meiosis. You must know that interphase is not part of meiosis. It's just a growth phase, because meiosis is regarded as a reproductive process in the reproductive cells, which are determining the cells which you call the gametes. So it includes DNA replication. That's where DNA replication takes place. Remember, when you're talking about DNA, we asked a question, where does it occur? We said in the nucleus. When does it occur? It occurs before the cell divides, or it occurs during interphase. So when you say before the cell divides, it means that interphase is not regarded as a phase of meiosis. It's just a growth phase. It's like saying that you give birth to a baby, and then the baby immediately cannot give birth to another baby. Needs some time for this baby to grow, so that now once the baby grows, then is ready to reproduce. When the cell divides and then goes to the last phase, it does not divide immediately. No, it requires some time for it to grow. So during that process of growth is what we call interphase. And the things which occur in that phase, some of them is DNA replication, doubling of the DNA. Chromosomes which are single-stranded, they become double. We saw it that we have what we call the unreplicated chromosomes. When they double, they become the replicated chromosomes. So when the chromosome has one thread, then we call it unreplicated chromosome, or what you call single-stranded chromosome. But when it becomes double, as we said, as you see it here, it becomes what it is called the replicated chromosomes, or double-stranded chromosome. So each chromosome will consist of 2 chromatids joined together by a centromere. So a chromosome, it must have a chromatid. It must have a centromere joining it. If it is replicated chromosome, it will have 2 chromatids, and these chromatids must be joined together by a centromere. Centromere is the middle part of the chromosome. Then DNA replication. What happens in DNA replication? DNA replication helps to double the genetic material, to double the chromosome number. As I said, that we don't say to increase the chromosome number. As we ask a question, it increases the chromosome number from what to what. So we need to say that it doubles the chromosome number or genetic material. So the material, so that genetic material, so that it can be shared equally by the new cells arising from the cell division. It means that now these cells, the new cells which are going to be formed, they are going to have the content. They are going to have the content. They're going to have the chromosomes. They are going to have other cell organelles equally, shared equally. because each cell must have this content. So if it doesn't have, then it means that it will be empty and then it won't be able to survive. So it is the function of interphase, the growth phase, to bring about the doubling of these organelles and the doubling of the genetic material, so that each cell, each new cell has a equal amount of the genetic material and also the cell organelles. If we have said that interphase is not the part of meiosis, then what are the phases of meiosis? Number one, we have what called the prophase. Number two, we have what called the metaphase. Number three, we have what called the anaphase. Number four, we have what the telophase. And then we go to prophase 2, prophase, metaphase 2, anaphase 2, telophase 2. So we are talking about prophase, metaphase, anaphase. What is it? What does it mean? Prophase means preparation. Pro means preparation. P versus P. Then you have metaphase versus middle. M versus M. And then anaphase versus apart. A versus A. And then T means telophase, T versus T. So even in prophase 2, metaphase 2, anaphase 2, and then telophase 2. So we form if you take the first letter, we form what we call IPMAT. This is in different color. It means that this interphase is not part of meiosis. That's why we don't have interphase 1 and interphase 2. We only have interphase. So it means that it's not a part of meiosis. So the process of meiosis begin with prophase, metaphase, anaphase, and telophase, whereby we say that this is preparation, middle, apart, and then they have reached the what? The terminal. What are those things which we are talking in middle, apart, terminal? What are they? We are talking about the chromosome, as you see them here. So this is trying to show you exactly what is happening. So it means that we start with one cell, the one cell divide from 2 cells, and then these 2 cells divide from 4 cells. But actually, what is really happening for this cell to divide is what you're going to look at in the details of each phase. Let's look at meiosis 1. Meiosis is divided into 2 phases, into 2 divisions, not phases. The first meiotic division and the second meiotic division. So in the first meiotic division, it will have prophase 1, metaphase 1, anaphase 1, telophase 1. And then the second meiotic division will have prophase 2, metaphase 2, anaphase 2, and then telophase 2. So the first meiosis, this is called reduction division, whereby when you talk about reduction division, you start with 46 chromosomes. At the end of meiosis 1, you end up with the 23 chromosomes in each cell. Hence, we call it reduction division. This division results in the reduction of the number of chromosomes from 46 to 23 in each cell. So the resultant gamete are haploid. It means that they have one set of chromosomes. From chromosome number 1 to chromosome number 23, they are not paired. It means that they are single, haploid, half. It means that the cell has 23 chromosomes, not 23 pairs of chromosomes. All right. Let's look at the phases of meiosis. The first phase is prophase, which means preparation. The cell is preparing for whatever is going to be doing. So the first thing is chromatin network. We say that chromatin network means that entangled DNA. DNA is in form. We don't have a direction when we look at it. We don't know where it starts or we don't know where it ends. So it is chromatin network. When it condenses, it becomes thick. When it becomes thick, it forms what we call the chromosome. So the chromatin network condense. When they condense, it means that now that it has formed the chromosome. Now these chromosomes, the chromosome which look alike, homo, which means the same, logos, which means structure. So it means that chromosomes which look alike, they are going to come together. That's why you're saying that homologous chromosomes are formed. It means that the chromosomes which look alike, they come together. When these homologous chromosomes, the way you see it here, when they come together, they cross over. What do they use to cross? They cross using the chromatids. The one chromatid of one homologous chromosome crosses with another chromatid of another homologous chromosome, and then the process continues. So the spindle thread starts to form. It means that now the spindle fiber, they start to form, and then once they start to form, the nuclear membrane starts to disappear. Let's look at the diagram here. So you see that here, the chromosome, they condense the homologous chromosome. You see this one is short, this one is long. So this short cannot go with the long one. No, the short goes as with the short, the long one goes with the long one. That's the principle. These chromosomes, they come together, and then the chromatids, they cross each other. When they cross each other, we call it crossing over. That's why I'm saying that crossing over takes place. And then the point where they cross over, we call it chiasma. We're going to look at this in detail. And then now these spindle, they start, the centrioles, they start to move to opposite poles. As they move, yes, as they move, the spindle starts to form. So once you see something like this, they have crossed like this, then you just know that yes, this is prophase. Why? Because the chromosomes are crossing over. Homologous chromosomes are crossing over. We don't have crossing over anywhere else except in prophase 1. Not in prophase 2. We only have it in prophase 1. Why in prophase 1? Because the chromosomes are still in homologous pairs. Let's look at the process of crossing over. So we have seen that the homologous chromosome, the chromosomes which look alike, they're going to line together. When they line together, one chromatid of each chromosome overlap. So it means that they cross each other. When they overlap, these chromatids, they must overlap based on the homologous pattern. For example, it means that the one chromatid from one homologous chromosome and another chromatid from another homologous chromosome, they cross over. And then when they overlap and they cross over, it means that they cross over at a point called chiasma. And then crossover takes place at a point called chiasma. If it is one, called chiasma. If there are many, we call them chiasmata. So when you say chiasma or chiasma, depending on the kind of English you are using, then chiasma or chiasma is a point where these two homologous chromatids cross over during prophase 1. And then now the chromosomes separate. So it means that now they have formed different chromosomes. I always ask this question to different people. Are you sure your father is your father? Why am I asking you that? You don't resemble him. Actually, you don't even resemble your mother. You walk, you go somewhere, and then you find out that you resemble someone. Why? And they ask you that, hey, when you resemble someone, I know that. Why? Basically, he might be your father. But because of crossing over, this made to the exchange of the genetic material. If you came from the same father, same mother, you breastfed on the same breast of your mother, why don't you look alike? It means that this crossing over, this process of meiosis helps in bringing about variation among organisms. So it is a very important process to make humans, individuals, different from each other. And then this helps in the survival. If all of us, we are the same, then it means that we won't be able to survive. Imagine everyone is rich. Do you think that there will be someone who is working for another one? Everyone has money. Do you think I would have taught you this? No, because you don't need this. No one would have been watching. You don't need this. You have your money. What else do you want? But because there is difference among these organisms, among us, then it brings about what we call competition. Hence, different people will work differently and in different environmental conditions. So that's the purpose of meiosis, to bring about variation among organisms of the same species. All right, let's continue with the process of crossing over. So these differences is being brought about by crossing over. Imagine a father producing over 100 million sperm for a single ejaculation. And then this sperm, each sperm is different from each other. What made them to be different from each other is the process of crossing over, because it forms different combination of chromosomes. All right, let's look at. So we're saying that you have one homologous chromosome and another homologous chromosome. They come together. Yes, when they come together, they overlap. The homologous chromatid overlap at a point called chiasma. Yes, if you look at ABC and then decide ABC, yes, when they cross, now this one takes this red and then the red takes the blue. That's why you see it here. The blue takes the red and then the red will take the what? The blue. These chromosomes during crossing over, scientifically we call it a bivalent or bivalent, based on how do you write that, how do you pronounce that, bivalent or bivalent. And then after that they separate so that each chromosome has genetically different, each chromosome has different material from one another. Look, if you see that ABC, it started with ABC, but this is a small letter. The small letter was found on this chromosome. And then this one, ABC, but this one is a capital letter. The capital letter came from this blue. If you look at it here, it came from the blue. It means that the blue, the red took the blue, and then the blue took the what? The red. So because of this, it's going to cause different gametes being formed. And then if you look at it, sometimes we can bring it in the exam like this. Homologous chromosomes, when they cross from top. So when it crosses from top, it means that you have to draw it on top. Don't draw it down. That's one of ways of losing marks if you don't show the variation correctly. So they have the chromatids cross over and then they exchange the material or the information. And then now the chromosomes now have new combination of the genes. Guys, don't forget that I told you that there is a gift in this video. Just click that link below. It will take you to a specific page on our website. And on that page, there is a code for you to obtain that airtime. Enjoy it. When you get it, when you load it on your phone, let us know that you got it. Thank you very much. Let's continue. And then you're saying that the second phase is metaphase 1. Metaphase 1, meta means middle. It means that now the chromosomes are always in homologous pair. Homologous chromosomes randomly align along the equator of the cell in 2 rows. It means that they align along the equator in 2 rows. One from chromosome number 1 to chromosome number 23. Another one is from chromosome number 1 to chromosome number 23. That's what it means. Each chromosome is attached to the spindle thread from each pole. This is what I'm trying to say. So they align in 2 rows. You see, they align in 2 rows, and then these chromosomes are being held by the spindle. See, each chromosome is attached to the spindle thread from each other. They do this arrangement randomly. So the key point is homologous because they're in pair and then each other. You see, this one cannot come here. No, they must be together like this. Homologous chromosomes. Yes, that's the key point. And then they come here randomly. Remember, we have 23 pairs, but we're just showing you 2 pairs. Yes, they are here randomly. So it means that another one can be here. This one can be on top. So this set can be on top. This one can be down. Or this chromosome can be this side. This one can be this side. That's what we call randomly. So when they separate, we're going to form different cells. So each chromosome is attached to the spindle fiber or spindle thread from each pole. That's what we are trying to show you, that these threads are attaching the chromosomes from each pole. So after that, once they are done with this, then they start to move apart, which you call anaphase. So in anaphase, the spindle thread shorten. They try to pull. When they shorten, they pull. It's like when you are a bodybuilder, when you carry something, the muscles shorten. When they shorten, they pull. Yes. So they pull the homologous chromosome to opposite poles. And then the chromosomes, what happens to this chromosome, the homologous chromosome pair, then they do what? They separate. The chromosomes of the homologous pair, they separate. It means that now they move to opposite poles. This is what is happening. So now they move to opposite poles. And then now, when they move to opposite poles, now they reach the pole. So in telophase, each chromosome reach at the pole of the cell, and then once they reach the pole of the cell, the cytoplasm and the cell membrane divide. When the cytoplasm and the cell membrane divide, that process is called cytokinesis. We call it cytokinesis, division of the cytoplasm. And then the two haploid cells are formed. Two haploid cells are what? Are formed, and then the nucleus or the nucleolus, that dense body in the nucleus, they reform. Then it reforms, and then lastly, the new nuclear membrane forms, and then now the cell appears to be a full cell. So this is what I'm trying to say. Yeah, so now we form two cells. Now these cells, cytokinesis took place, and now the nuclear membrane start to reappear, and then the cell goes back as if it is in prophase. So now when it reaches there, now each cell, each of it will undergo meiosis 2. It means that now this one is going to go under meiosis 2, even this one is going to undergo meiosis 2, so that you form 2 here and 2 here. So when you add them, you form 4 cells. So when you go to meiosis 2, what happens? Now prophase 2, you go to prophase 2. When it's meiosis 2, prophase, it becomes prophase 2. The 2 must be there. If you don't write it there, we won't give you a mark because we don't know which prophase is it. So prophase 2, what happens? The nucleolus, remember we said that it appeared, reappeared. So now the nucleolus and the nuclear membrane, they disappear again. But now here we don't have crossing over. So the new spindle fibers are formed. Yes, they start to form, and then the chromosome become visible. It means that they condense, and then it becomes visible. And then now, once they become visible, now it's ready to undergo metaphase. They go to the middle of the chromosome, the cell. So now you see it here. Now it is forming like this. And then now they are ready to move this thing to attach themselves to the chromosome. But now we don't have homologous chromosomes. We have chromosomes which are not homologous. It means that crossing over cannot take place because it already took place before. Yes. So now in metaphase 2, the individual chromosome line up. Here, individual or one chromosome align along the equator singly or individually. So the other side, we say that chromosomes, they align along the equator in homologous pair and randomly. Here, they align along the equator singly or individually. So you have to know the difference. So sometimes you ask you the difference in meiosis 1 and meiosis 2. You can use the process to differentiate between the two. So at the equator of the cell, with the centromere attaching the spindle fiber, or centromere attached to the spindle fiber. And then now, this is the picture. If you rotate it, you will see that these chromosomes are in the middle. You see, they're in the middle. If this is the pole and this is another pole, they are in the middle. So this one becomes the equator. Remember, I said that equator is an imaginary line. It's an imaginary line. It's a line which we just imagine, but the line is not there. It's a imaginary line which divide the cell into 2 equal parts. It's the same thing, equator of the earth. Imaginary line running from east to west and dividing the earth into 2 equal parts. We're just imagining, but the line in actual sense is not there. We just imagine that that's the center of the earth. So we just imagine that's the center of the cell. So in anaphase, once they're in the middle, they have to separate to opposite poles. So in anaphase 2, you have spindle fibers start to form. So the spindle fibers, they contract. When they contract, it means that now they pull the chromosomes apart. But now, because they are single chromosomes, they are not homologous. Now it's this the chromatids which are going to separate. Remember, chromosome is divided into 2 chromatids. Now the one chromatid is going to go one side, and another chromatid is going to go to another side of the cell. So this is what I'm saying, that now the chromatids move to opposite. It was together here. Now this one pulls it this side, and this one pulls this side. Check the direction of it's not facing this way, it's facing that way, because the force of pulling is coming from this side. So that's why it's supposed to be like that. When you're drawing, make sure that you take caution when you're drawing it, so that you draw it properly. And check about the shadings. Also, the shadings are very important when you are doing it. The daughter chromosomes reach the poles once in telophase 2. In telophase 2, the daughter chromosomes reach the poles. When they reach the poles, we don't call them chromatids anymore. We call them chromosomes. Actually, That's what we call single-stranded chromosomes or unreplicated chromosomes. So it's supposed to get time so that this time allows these chromosomes to grow so that now it becomes double-stranded again. That's why we need what you call interphase. So a new nucleus then forms. It means that the nucleolus also come back, or it means that it reforms. Then the cell membrane of each cell constrict, and then the process of cytokinesis occurs. When the cytoplasm divides, now the process of cytokinesis occurs, and then for haploid, it must be haploid. Daughter cells are formed, and then now each daughter cell have the chromosome number to that of the parent. So that's why you're saying that meiosis is the process whereby one cell divide to form 4 daughter cells which are haploid, or which have half the chromosome number to that of the parent. And these chromosomes are genetically different because of the crossing over which took place. So the daughter cells are genetically different, as I've just said, and then these are the cells which will form. We form 4 daughter cells, and then you see, we don't call these ones chromatids, we call them single-stranded chromosomes. Watch out for this before you write your SBA. So the importance of meiosis. What are some of the importance of meiosis?
Welcome back to our class. Yeah, today we are going to be looking at the process of protein synthesis. What is protein synthesis? So protein synthesis is divided into 2 words. Number 1, it is the protein, and the synthesis. When you talk about synthesis, it means manufacture, production of proteins. Protein synthesis is the process by which proteins are made in the cell of an organism to form enzymes, hormones, and the new structures of the cell. So your body is made up of different structures, and these different structures are made up of protein. So it means that if you don't have protein synthesis in your body, then it means that you can't grow, you can't have these hormones, you can't have these enzymes. So the marking point here in the definition is the word proteins are being made in the cell. This process is divided in 2 different phases. The first phase is called transcription. And when you talk about transcription, it means formation of messenger RNA. Then number 2 is translation. Translation is the formation of a polypeptide chain. The difference between polypeptide chain and the protein is almost the same, but it depends on how many amino acids are there. We are going to see the amino acids in detail. So how many amino acids are there? If more than 50 and above amino acids, then we call it a protein. But if it's less than 50 amino acids, we call it a polypeptide chain. This diagram shows the whole phase of protein synthesis. This is the nuclear membrane. These are nuclear pores. So we have gone into the nucleus, and then we have gone into the cytoplasm. So this diagram you see, it's just showing you the nucleus and then the cytoplasm. We know very well that the DNA is found in the nucleus. And then this DNA, because it is a king, is the one which codes for the protein, doesn't move out of the nucleus. It's like a king who doesn't move out of the palace. He has the servants, and these servants, they go take information what the king has said. So these messengers, the transfers, they act as servants for DNA, because DNA wants to produce a certain protein. So it will send a messenger, and then the messenger takes the information to the cytoplasm, then the transfer RNA will bring the amino acids. So when they bring a diagram like this, you have to know how to label. That's the first point you need to know so that you can pass this question. The first thing is you have to know that this is DNA. When it is coiled, if you see something coiled, is DNA. If it is too coiled, then we call it chromatin network. Then the DNA is done which unwinds, it forms the messenger RNA, and then the process of formation of messenger is what we call transcription. Then this messenger RNA will move out of the nucleus via the nuclear pore, and then it goes to the ribosome. You see, these are the ribosomes. This messenger will align itself on the ribosome, and then the transfer RNA will bring the amino acids to this ribosome, and then a chain of protein, a chain of amino acid is formed. Therefore, this process whereby the messenger brings in and then the transferrin brings in the amino acid also, the chain is formed, is called translation. So the major difference between transcription and translation: transcription occurs in the nucleus, while translation occurs in the cytoplasm. So you have to know at least the basics of this process. Before we go to the process of transcription, so this is a simple diagrammatic representation of the whole process of protein synthesis. So you expect DNA to unwind, and then when it unwinds, it forms it exposes two strands, but one strand will act as a template for formation of a new strand, and then this new strand is what we call the messenger RNA. This messenger RNA, we saw that it moves out of the nucleus via the nuclear pore, and then it goes to the ribosome. The moment it reaches the ribosome, then it starts the process of translation. Then amino acids are joined together, are brought by the transferrin A. They are joined together to form a polypeptide chain. So if the number of amino acids become a lot, the more than 50 and above, then it forms what you call a protein. So the protein, it must have is a heavy molecule compared to a polypeptide chain. So now let's look at what happens in transcription. We see in the exam, what do you write when we find such questions, and then what happens in translation. Transcription. Don't forget, this is a recall that transcription occurs in the nucleus. You have the DNA, and then you have the DNA to unwind, and then messenger RNA is formed. Then messenger RNA moves out of the nucleus by the nuclear pore, and then it goes to the cytoplasm. Basically, that's what you're going to be explaining. So the first point, what you see in different color, it means a marking point. DNA double helix. You have to tell us the natural shape of DNA. DNA double helix unwinds. So that's the first step. Number two, it will say that when the weak hydrogen bonds break. Here, when you say weak hydrogen bonds break, or you say DNA double helix unwind, then it unzips. It's the same as saying weak hydrogen bonds break. What's the meaning? If you look at your trouser, you try to open it, you zip down. When you zip down, you are breaking the bond which we are holding the zip together. You understand? So when you say when you unzip, it means that you are breaking down the bonds which are there. That's why we say that weak hydrogen bond break. Or you can say DNA double helix unwind, then unzips. So you get the same mark. One strand will act as a template. There is a major difference between DNA replication and transcription. Here, when you're talking about transcription, one strand is the one which acts as a template, while the other side, the two strands will act as a template. So something which look like a mirror, so that there is one strand, so that the new strand is being formed. Then to form the messenger RNA, one strand will act as a template, as you see it here, and then messenger RNA is formed. It uses free-floating nucleotides. When you say this, you are supposed to specify, because when you say free-floating nucleotides, in the nucleus, there is free-floating nucleotides of DNA and free-floating nucleotides of messenger RNA. So you have to be specific when you come to this. You have to say that free messenger RNA nucleotides. Yes, so these free messenger RNA nucleotides which are floating in the nucleoplasm are the one which we use. Yes, so if you just say using nucleotides, we'll cross it, because we have different kinds of nucleotides in the nucleoplasm. So you have to be specific that free messenger RNA nucleotides. Then the nucleotides, the messenger RNA is complementary to DNA. So it means that where there is A, you're going to have U, and where there is C, you're going to have G. Remember, the messenger RNA does not have thymine. It has what you call uracil. That's why we say A goes with U and then C goes with G. Then the messenger RNA now has coded message of protein synthesis. It means that now it is ready to move out of the nucleus and then go to the cytoplasm, so that it can take the information DNA wants the protein to be manufactured. Yeah, that's why you're saying that now the messenger has the coded information for protein synthesis. Then we are saying that the messenger RNA moves from the nucleus through the nuclear pore, and then it goes where? To the cytoplasm. Where exactly in the cytoplasm? That is on the ribosome. So it basically, this is the general process of protein synthesis, basically that is transcription. So what you see in different color, it means the marking point. DNA double helix unwind, weak hydrogen bonds break, and then one strand will act as a template, and then for formation of messenger using free messenger RNA nucleotide in the nucleoplasm, and then the messenger RNA is complementary to DNA, whereby A goes with U and then C goes with G. And this messenger which is formed has a coded information for protein synthesis, and then this messenger moves out of the nucleus by the nuclear pore to the ribosome. Basically, that's what we want. 6 marks from here. In most cases, you see it as a short question which requires you to write these statements the way you see it. Please don't change. If you change, make sure that the different colors don't change, because those are the marking points we are looking for. And then let's look at the process of translation. Here, let's look at the process of translation. Welcome back to our class. Don't forget to subscribe if it is your first time to visit our channel. What is speciation? Speciation is the formation of new species. So what is a species? We saw it time before. We saw it that a species is a group of organisms which can interbreed to produce a fertile offspring, like which process leads to the formation of this new species. So let's look at the process which results in the formation of new species. So here you have a population. This population is population 1 and population 2. These two populations, they can interbreed. They have the same kind of species, but now the geographical barrier has come in. So we are saying that a population of or a particular species may be split into 2 by a geographical barrier. So this was, for example, it was one population. Actually, it was one population. There is gene flow. The gene flow means that they can interbreed, they can have sex, they can meet. Yes, for example, population of humans in South Africa and the population of humans in America. Though the distance is great, but you have planes which can go there. You have sheep which can go there. Still, we can interbreed to produce what? Though we different populations, but still we have the same species. Why? Gene flow is still there. So the population can be separated. We are saying that can be split into 2 by geographical barrier. Geographical barrier could be a river, mountain. It could be even a box, depending on which kind of species are you talking about. It must be a barrier which is separating them. If it is in the environment, then it could be environmental barrier. If it is in a lab, like bacteria, it can be just a small box. Yes, so it depends on how big is the species. So now we are saying that as a result, 2 parts of population cannot interbreed. So now a geographical barrier has been introduced here. Yes, and then now these 2, they cannot interbreed. There is no gene flow. There's no gene flow. This one cannot have sex with this. Why? Because of the geographical barrier. Therefore, there is no gene flow between the 2. It means that these ones cannot cross this side and these ones cannot cross this side. Yes, and then you're saying that natural selection occurs independently. So because of the different environmental condition this side and different environmental condition this side, then natural selection will occur differently because of the difference in the conditions of the environment. So you're saying that this is due to different environmental conditions. Yes, it means that natural selection will occur differently because of the different environmental conditions. The different environmental conditions will result in natural selection occurring independently. And then they're saying that as a result of 2 populations become, the 2 populations become genotypically and phenotypically different. over a period of time. So as many generations pass, due to this geographical barrier, they cannot interbreed. They start to change, and then these ones will be wiped out because they're not being favored by the environment. The red one will survive. Also, these ones will be wiped out. The blue one will survive, and after so many generations, you'll find out that they will become genetically and phenotypically. Genetically, in terms of gene, and phenotypically, the way you see them, different colors now. Yet they were the same color. They become different over many generations because of the genes have changed. Then automatically, even the phenotype must change. And then if you remove the barrier, we are saying that even if even if the population mix at a later time, yes, if you remove the barrier and you leave this population to mix, yes, what will happen? They will not be able to interbreed again to produce a fertile offspring. They won't be able to interbreed to produce fertile offspring. Some of them they can interbreed, but they may not produce a fertile offspring. We will see this one as we come in. So what does it mean? It means that one or both of the population have become new species. In this case, the two populations have become new species. Why? If you look at the original kind of species, is different from what is being formed. So that's how you can explain speciation. But when you are bringing in the exam, we might not bring something like this. We can bring it like this, or we can bring something different. We have given you an example. We give you an example whereby you're supposed to apply what you learned. Yes. So if the example is talking about elephants, you must explain based on elephants. If you're talking about maybe bacteria, you must explain based on bacteria. Birds, any kind of organism they are brought, as long as you talk about geographical barrier, you have to tell us which geographical barrier is there. Two, they cannot interbreed, yes. There is no gene flow, yes. You must talk about natural selection and occurring independently, and you must talk about due to the environmental conditions. So you must talk about the genetically different, resulting in phenotypically different organisms, and then you must talk about when you remove the barrier and leave them mix, then you won't be able to interbreed to produce a fertile offspring. Then you tell us how many have become new species. Then you'll be able to get the mark. Describe how these two species of the flies were formed. So now it means that now they have brought an example of flies. How did they form? If you look at these flies, as if they're in the lab, yes, they are being separated by the what? Box. At first, it was one population, as we told you. It was one population. Now the population is separated by the box. Yes, the geographical barrier now. The barrier here is now the box. This one, there is no now gene flow. You see that they are still the same kind of species. So there is no gene flow between them. These ones cannot come here and then interbreed with this. And then because of there is no gene flow and they are being separated differently, then they have different environmental condition. In this case, what is the different environmental condition? Is the kind of food which is present. Here we have starch food, and then here we have maltose. So it means that they have different environmental conditions. So those which cannot feed on starch, they will do what? They will die. And those which can feed on starch, they will survive. While here, those which cannot feed on maltose will die, while those ones which can feed on maltose, they will survive. Yes. So over many generations, yes, it means that due to no gene flow between the two populations, yes, then this will result in a different genotype. This will lead to these species becoming genetically different and phenotypically different due to the different environmental conditions as a result of natural selection occurring independently. So the different genotype resulted in different what? Phenotype. And if you remove the barrier and you leave them mate, they cannot interbreed to produce a fertile offspring. So it means that now this and this, they have become new species. So that's how you can explain, depending on the question they have given you. You have to be keen. You have to look at which barrier, which kind of environmental condition, which kind of species have been given. So, and then how many have become new species. If you look at them, yes, you'll find out that they are different species. If you look at this, is different from this, and this is different from this, then it means that the two have become different species. There was one population of flies. The population of fly species became separated by the wall of the box into population A and B. Yes, we saw it. And then because there is no gene flow between the two populations separated, yes, what happened? Natural selection occurs independently in each A and B due to exposure to different environmental conditions. In this case, that is the food source. The food was different. So because the food source was different, this one fed on maltose, and then the other one fed on what? On starch. So that the environmental conditions were different. Hence, this will result in the population becoming very different from others. That is genotypically and then phenotypically. You have to start with the genotypically and then talk about phenotypically, because it's the genotype which determines, the phenotype. And then you're saying that when the population were allowed to meet again, they were not able to interbreed. So when you remove the box and then make them mix, they are not able to interbreed to produce a fertile offspring. Don't forget this. And then you're saying that the population A and B are now new species. So they have become new species. Yes. So P means a point or a mark. It could be. Yeah. So it should not confuse you. So basically, that's it. And then you're saying that another question is this. You can also apply it in this case. Say geographical barrier, they have not talked about it. You see that it was one population divided. You see now it starts to change, then finally changes, and then it forms different two new species. So it's almost the same as the previous one, but now it is in a different angle. Yes. Try to download these slides and then you can try to practice such questions. Yes. So what are some of the mechanisms of reproductive isolation? When you talk about reproductive, it means that sex is not happening. Yes, it has been isolated. Organisms have been isolated from sex. That is reproductive. They are not reproducing. It results in them not reproducing. So that isolation, what are some of the mechanisms? What are some of the things which result in these organisms not reproducing? Keeping species separate. So number one, breeding at different times of the year. Yes. So this one breeds in fall, and then this one breeds in late of the winter. So it means that they cannot have any time when they are breeding at the same time. So they are being separated by the different times of the year. Number two is species-specific courtship behavior. Like the way how you do your courtship is different from how dogs do the courtship, is different from how the birds are doing the courtship. So it is different. We have different ways in which we do our courtship. We seek for sex differently. We have different ways. So that one can also, the way how you do it and the way monkeys do it, it must be irritating you, and yet the monkeys are enjoying the same story to humans. So if it happens the way how we do courtship, then it means that it could lead to isolation. Adaptation to different pollinating agents or pollinators. You see that here, this is an insect, this is also insect, but it is a butterfly. This is another insect, kind of different insect. You have different insects. This is a bird, this is a bat, this is a human. So they have different pollinating agents or pollinators. So because of that, they are not, if this cannot come, then it means that you will not carry out pollination, or it will not reproduce. So because of that, they are being isolated, or they are being separated. Then you have yes, you can have sex, you can have what, you can produce a baby, but you end up producing an infertile offspring. For example, a horse which is 64, so when it is one end, when meiosis takes place, it is 32, and then a donkey which is 62, 31, when it divides, then they produce 32 plus 31 produces what? Produces 63. So when it is going to carry out meiosis, what is going to give? It is not applicable. Then it means that this one will be it won't be able to reproduce. So it is infertile. They have crossed, they have produced a baby, but the baby is not fertile. Yes, so it cannot reproduce again over and over and over again. So it becomes still where they are being separated. And then lastly, prevention of fertilization. So you hear that a person produced a dog. Does it mean that that person slept with a dog? No, it doesn't mean like that. It could be mutation. But now, is it that if you produce a snake, produce a dog, or produce whatsoever, it's not that you slept with the dog. It's because we have different kind of sperms and ova. Yes, so fertilization will not take place. You see, the egg of a human and the egg of the bird, the chicken, they are totally different. Yes, humans are big, but their eggs are small, while birds are small, but their eggs are big. So there's no way how you can reproduce. These are some of other examples of speciation. How did they form these beaks? Here is another example which tries to how did these two species came about. The question can come, how did they come about? All these are other examples of speciation. So you can read about them or just have one the way I've explained it, then you'll be able to apply others.
The theories of evolution. This is the core. Now it's like we are starting what you call evolutions. Number one. The first one is Lamarck's theory, which we call the Lamarckism. This guy was there in 1744 to 1829. No one was born by this time, and no one was thought of. His theory, if you look at it, many people try to criticize it, but for me and the other scientists, they see that his theory was very important because it gave us a benchmark, how life is evolving from different organisms. Number two is Darwinism, or Darwinist theory. Charles Darwin, from 1809 to 1882. These guys, they did a great job. If they didn't do the great job they did, we wouldn't have seen that evolution. Why is evolution taking place the same way it is happening? So let's look at the Lamarckism. This guy was a Christian, like a bishop. Yeah, this guy was very clever because he started the story. Okay, this guy says that. Number one, this guy Jean-Baptiste de Lamarck started his life from 1744 to 18 29. How many years are those? Almost 70 something years, I think so. If I'm not mistaken, I'm not a mathematician. This guy Lamarck used the laws, two laws. The first law is the law of use and disuse of a structure. Law of use and disuse of a structure. Number two, he also used the law of inheritance of acquired characters. So he said that in the law of use and disuse of a structure, organisms, an organism uses a structure or organ more frequently, it becomes bigger and developed, becomes bigger, or it becomes better developed. If you use the structure, it becomes more developed. The more you use the structure, the more it becomes bigger and stronger. And then that is the use. If you use the structure, it becomes bigger and stronger. If you don't use the structure, it becomes smaller and finally disappears. Quote me right. Not all structures. There are some structures which you don't need to use and disuse. Just make them disuse. Don't use them. The more you use the structure, so you have to use the structures. That's what he says. The more you use the structure, the more it becomes bigger and stronger. If you don't do that, it becomes smaller and then finally disappears. So he says that as organisms uses structure or organ more frequently, yes, the more you use the structure. This, at first it was like this. So now this started. The grass was low. So the one this grass, if the grass was like this giraffe will not develop its neck. But if the grass was like this and then the drought came up and then you find out that the only grass is at, or the foliage is at on the top of the tree, so now the giraffe starts to stretch their necks. So when they stretch the neck, as they stretching the neck becomes longer and longer and longer. So it's using this part. It's using this part so that at least the neck is growing and growing. And then this character does not stop there. It is being inherited to the next generation. So if you see it, says that the structure is being used. If you look at this, the structure is being used frequently as it gets the food on the top of the tree. So frequently, many times, so it becomes better developed. So it becomes longer. If you look at it, this and this, so now it becomes longer and then it becomes bigger. So that now the offsprings, they have the same kind of character. For example, if you're a bodybuilder, yes, and then you make muscles, you want to have a kid, the kid is going to be born with the muscles. The muscles, the kid doesn't need to go to the gym. No. He's going to be born with the muscles. The more you use the structure, the more it becomes bigger and stronger. And then these characters are being transferred to the next generation. That's what he says. However, we will see that even you see that there are some loopholes which are there. But it gave us at least a basis how to explain these ideas. And then we are saying that if the organ does not use, now that this is the disuse, this is the use, this is the disuse, does not use a structure or organ frequently, they become smaller, less developed, or reduced in the size and may disappear altogether. So if you remember when you're talking about the theories of evolution, we talked about embryology, and then we say that we had a tail. And even if you go to birth, you find out that you had a tail. If you try to check behind your, you know what I'm talking about. Yes, you find out that you have a tail. But because we didn't use that tail, it became smaller and smaller and then disappeared. That's how Lamarck is trying to explain that if you don't use the structure, it becomes smaller and smaller and finally disappears. So that is the law of use and disuse. And then the law of inheritance of acquired character, that's the number 2. He says that characters developed during the life of an individual can be passed on to the next generation. For example, like now you see that these giraffes, what happened? The giraffe was stretching the neck. So this giraffe will give rise to this. This neck will be longer as is longer, so it passes the genes from here again to the next generation. The characters which you obtain during lifetime, you pass them to the next generation. If you get an accident and then the leg is cut, then you're gonna produce a baby who is not having a leg. That's what he says, that the characters you obtain during the lifetime, you pass them to the next generation. So that is Lamarck's theory. They can ask you, describe how Lamarck explained how did the giraffe came up with long necks. Describe how Lamarck explained how tigers came up with stripes. Describe how Lamarck explained how elephants came up with long trunks. You can use law of use and disuse, and then the inheritance of acquired character. Make sure that when you're explaining, you have those two there. Here is a question. Let's look at it. Account. Write an account showing how Lamarck would explain the development of longer necks in the modern giraffe. 5 marks. Here is the memo. Number 1, all giraffe had short necks. We saw them that all of them had short necks originally. That's how we mark now. We give you a tick. All giraffes had short necks. It means that there was no variation among these giraffes. All the giraffes had short necks. That's number 1. Number 2, these giraffes frequently stretched their necks. As they are stretching their necks, yes, they are trying to reach the food on the trees. Yes. Number 3, they did this to reach the leaves that were available only higher up on the trees. So as they're stretching their necks, they are trying to get the food on the trees. As a result, their necks became longer. Their necks became longer. Yes. And then the characteristics now, that is the law of use. From here up to here, that is the law of use and disuse. Yes. And then now, the giraffes have obtained the long necks, but you want to pass these characters to the next generation. He says that the characteristics of the long necks acquired in this way were passed on to the next generation. So this point proves the inheritance of the acquired characters. So all giraffes had short necks, meaning that there was no variation. Number 2, the giraffes frequently stretched their necks. As they stretch their necks, they did this. Why did they stretch their necks? They did this to reach the leaves that were available on higher, only higher up on the trees. And then as a result, the necks became longer. Yes. And then these characteristics are meaning that the long necks acquired during lifetime were passed on to the next generation. So now, after that, eventually all the giraffes had long necks. That's how he explained how did the giraffe came up with long necks. But if you look at Darwin's theory, he explained it using a different angle. But there are some points, at least, which are a little bit similar. It means that Darwin used the idea of Lamarck to at least to develop the theory of Darwinism using natural selection. Explain the Lamarck's theory. Why? Explain why Lamarck's theory was rejected. Explain. If you look at it, do you think Lamarck's theory is correct? It could be correct in some instances. Is it that when you use the structure, it becomes bigger and stronger? Yes, some structures, they become bigger and stronger. If you look at the mouth, yes, every day you use your mouth, does it become bigger and stronger? Does it? Oh, if it becomes bigger and stronger, then it means that his theory is right. But if you look at the people who are doing the muscle building, if you go to the gym, start to use the muscles, you do the activities, do you have the muscles? Yes, the more you use the structure, the more it becomes bigger and stronger. But do you pass on all the characters you obtain during lifetime? No, no, you don't pass all the characters. The physical, the phenotypic characters you obtain, you don't pass them to the next generation unless there is a mutation. So basically, that's it. We are saying that he says that number one, there is not enough evidence to show that acquired characteristics are inherited. It's not that when you obtain a character during your lifetime, then you pass it to the next generation. It's not like that. Number two, organisms do not evolve because they want to evolve. Lamarck says that if the giraffe wants to get the long neck, so just has to stretch, then it becomes longer, the neck becomes longer. If it wants to have a shorter neck, then the neck becomes short, then you get. If you want to become tall, you just have to jump like a frog, and then you become tall. So if you are tall and then you want to become short, you just have to squeeze yourself, then you become. So that's what he's saying that. And then you produce kids who are tall, or you produce kids who are short. If you come, you want a character, you just have to practice it and then you get it. So we say that no, organisms don't evolve because they want to. No. They just have to evolve because of natural selection. Then number two, organisms don't evolve because they want to evolve. No. Organisms evolve because of other factors. So basically, that's it. Okay, they're saying that Darwinism. So we have seen Lamarckism versus now Darwinism. Let's look at Darwinism. We say that Charles Darwin is one of the great scientists. So this guy existed in 1809 to 1882, almost still 70-something years. So these guys, they stayed longer, né? They stayed longer. Okay. His theory is based on natural selection, survival for the fittest. Guys, this is natural selection, and natural selection is happening everywhere. Survival for the fittest. I repeat, his theory is based on natural selection. Natural selection means survival for the fittest. The best suit in an environment will survive, and those which are not suit will die, and then they will extinct. So now we are saying that organisms, he says, his theory, which is based on natural selection, it goes like this. He says that organisms produce a large number of offspring. You see, check these rats. They produce a large number of offspring. Have you ever asked a question that you have never seen butcheries for dogs, but you see butcheries for cows? But cows produce one kid or offspring per 9 months. Dogs, they produce 6 or 7 per 6 months, for example. But why is it that every day you slaughter cows, a lot of them? Every day you don't slaughter dogs. Why is it that you don't see a lot of dogs, but cows are a lot? Why is it like that? So we are saying that yes, though dogs produce a large number of offsprings, why is it that they are not abundant everywhere? There must be other factors. It means that natural selection must happen or must apply on these dogs so that the number are reduced. You have a lot of rats. Produces a number of rats, but where are the rats? You don't see them everywhere. There must be some factors which can which bring about the reduction in the availability of these rats everywhere. So we are saying that organisms produce a large number of offspring. So if you don't want to produce, make sure that you produce. So okay, there is a great deal of variation among the offspring. So look at this. This one, you see this color, is another color, is another color, is another color. All these are coming from the same father, same mother, but different characteristics, meaning that there is variation among organisms of the same species. So number one, organisms produce a large number of offspring. That's number one. Number two, there is a great deal of variation among the offspring. Number three, he says that some have favorable characteristics and some don't have. Some have favorable characteristics, yes, meaning that those characters, they can help them to survive. Some don't have favorable characteristics. There are so many kids who want to join our class, but they don't have favorable characteristics. They don't have the data. Maybe they don't have the nice phone to connect, but others do have. Those who have, they have connected and they're in class with us, even if they are home. And those who don't have, they are not in class. So they are wiped out. That is what we call natural selection. So now we are saying that when there is a chance in environmental condition, so when the environmental conditions changes, so when there is a change in environmental condition, or even competition, there is competition for food, for example, competition for space. Yes, what happens? Organisms with a favorable characteristic will survive. So the organisms with a favorable characteristic will survive. And organisms without favorable characteristics will not survive. So it means that those without favorable characteristics, those with unfavorable characteristics, they will die. And the organisms with favorable characteristics survive, while organisms without favorable characteristics, they will die. Those which have nice phones with internet, they will connect, they will survive, and those without, then for them they will die, meaning that they will be wiped out from the system. They won't study. You see now how natural selection does? Those who are favorable, they continue with life. And then they're saying that organisms that survive reproduce, because now they have the chance to survive. Then they reproduce, and then they pass on the allele to the next generation. So that it means that favorable characteristics will make people to survive and reproduce, and then the characters which are good will be passed to the next generation. So I repeat, he said these are the observations he used. Number one, organisms produce large number of offspring. So whenever you explain, you must include large number of offspring. There is a great deal of variation. Whenever you explain this, you must talk about this great deal of variation. So in this case, if they say that giraffes, explain giraffes, you will talk about variation. Variation, some have long necks, some have short necks. Some have stripes, some have no stripes. So that's the variation. Some are big, some are small. That's the variation. There is a great deal of variation among the organisms. Some have favorable characteristics and some don't have. So for example, if I want a giraffe with long neck, those which have long necks, they will survive. They have favorable characteristic. That's the characteristic the nature wants. And then if there is a change in environmental conditions whereby the food is only on the top trees, yes, what happens? Now the giraffe with the long necks, which have the favorable characteristics, they will survive because they will eat the food from the tall trees. And those with the short necks, they will die because they won't have what to eat. They have unfavorable characteristic. And then you are saying that giraffes that survive, yes, those which survive will reproduce in numbers. They will reproduce and then they pass their genes to the next generation. It means that now the giraffes with the long necks will pass their genes to the next generation because they have food. When we come back, we are going to look at this question, account or write an account showing how Darwin would explain the development of the long neck in the modern giraffe. How, yes, we saw how Lamarck, for the use and disuse of the structure and inheritance of acquired character, use those laws to explain that. But now Darwin used the concept of natural selection. Those with the favorable characteristics will survive, and those without favorable characteristics, they will die. So how did he use this concept to explain this?
Welcome back to our class. Yeah, today we are going to be looking at the process of translation. Here, let's look at the process of translation. Basically here, now the messenger has moved out of the nucleus. It has gone to the cytoplasm, and then it attaches itself on the ribosome. So now we want to bring the amino acid to the ribosome so that a chain is formed. Number one, what you say is each transferrin A carries a specific amino acid. It means that it has 3 bases. These 3 bases, they code for a specific amino acid. These bases are called anticodons. When the anticodons on the transferrin A, yes, those 3 bases happens, matches the codons on the messenger. So the 3 bases on DNA, we call them codes. The 3 bases on the messenger, we call them codons. The 3 bases on the transfer, we call them anticodons. So the DNA code for the codons on the messenger, and the messenger codes for the 3 bases on the transfer, which are the anticodons. And then the anticodons, they code for the specific amino acid. That's why we're saying that each transferrin A code for a specific amino acid. It's like a key. The other one will not enter or to open. When the anticodon on the transferrin A matches the codons on the messenger, so what happens? This transferrin A will bring the required amino acid to the ribosome. So it will bring the required amino acid to the ribosome, and then the amino acids become attached by a peptide bond. We call it a peptide bond, not polypeptide bond. Don't say polypeptide bond, we will cross it. It's called a peptide bond. If there are many, we call them peptide bonds, not polypeptide. When you say that, cross it. So amino acids become attached by peptide bonds, and then to form the required proteins. That's what we want. So this is what exactly what is happening. This is exactly what is happening. Each transferrin A carries a specific amino acid. For example, it carries a specific amino acid, a specific amino acid. This cannot carry another acid, no. And then you're saying that when the anticodon, when these 3 anticodon, yes, match with the codon, yes, match with these codons. For example, is T goes with A, T goes with A, and then A goes with U. When they match like that, yes, now what happens? This transferrin A will bring this amino acid, which these 3 bases, yeah, which code the anticodon, will match this. So it brings this required amino acid to it brings the required amino acid to the ribosome. So now the amino acids are joined together. So it keeps on bringing, and then the amino acids are joined together by these bonds, which you call, you see, these bonds are called the peptide bonds to form a required protein. And then you form this polypeptide chain, but now it grows. Once it grows, it is more than 50 amino acids, then we call it a protein. So these are amino acid. If they ask you what is this, it is called a polypeptide chain. Yes. And then this is the messenger, the 3 bases on each, we call it the codons. And then on the DNA, we call it the codes. And then on transfer, we call it anticodons. And then this molecule where transfer, where messenger lines is up, is called a ribosome. much for watching. Next time we are going to be looking at the mutations on DNA. Mutations on DNA, and then we're going to see some questions. How do we answer questions concerning about DNA, the structure of DNA, so that you are able to pass this topic. Just 1 week is enough to grasp this topic. So you need to know that this is DNA, this is messenger, this is messenger still, this is transferrin A, this is a polypeptide chain, this is amino acid, and then this is a transferrin A, it is a T, that's why it's called T, and then this is 3 bases, which we call the anticodons. 3 bases on this messenger, we call them codons, and then 3 bases on DNA, we call them codes. Yeah, thank you very much. Welcome back to our class. Don't forget to subscribe if it is your first time to visit our channel.
We have 3 types of dominancy: complete dominancy, codominancy, and incomplete dominancy. So let's look at them in detail. When we talk about complete dominancy, when you are dominant, it means that you have powers over another individual. So it's a genetic cross where dominant allele masks, blocks the expression of the recessive allele in a heterozygous condition. So in complete dominancy, you have heterozygous condition. So you have what you call the incomplete. When we talk about incomplete, it means that yes, the dominancy is there, but it is incomplete. So it means that one brings 50 and then another one brings also 50. So you're saying that it's a genetic cross where none of the allele is dominant over the other. Neither of the allele is dominant over the other. It means that they have equal powers. The president of South Africa and the president of America, both of them, they have the same powers in their countries. So it means that neither of the president is dominant over the other. When it is the meeting, the president of South Africa will have the same contribution like the president of America. That's the meaning. Not that, no, because America is the superpower, now South Africa, no. Both of them, they are presidents. Neither of the two is, they are dominant, yes, they are dominant, but neither of them is completely dominant over the other, over each other. Although your country is big or small, still you have the same power as presidents. For example, you have white and red. When you cross them, they form what called the intermediate. Intermediate means that neither of the two is dominant over the other. When you cross them, they produce the intermediate. It's not white, it's not red, but it is in between there. It produces a new character. So the character which is being produced is new. That's the meaning. Yes. But here you have the one character from the parent. Black and white, you either produce white or you produce black. Here when you cross, you don't produce white, you don't produce black, but you can produce gray, which is in between the two. It's not white, it's not black, but is in between. That's the meaning, intermediate. And then you have the co-dominant. Co means together. Co means together, means two things are existing at the same time. They have the same power, they have expressed themselves at the same rate. For example, you have white and black. When you cross them, you produce a mixture of white and black. They are co, they are together, they are expressed outside or in the phenotype together. You have the white and the black, they are living together. That's the meaning of co-dominance. It means that both of them are dominant, and their dominance is being expressed in the phenotype at the same time. So here you're saying that a genetic cross in which both alleles are expressed equally in the phenotype. Let's look at these examples. You have complete dominancy. Dominance allele completely masks the recessive one. It means that you have here, you have the red and the pink, and then you produce red. It means that you have one character being produced in the offspring, either red or pink. One must be produced. Then in incomplete, you have red and white. You don't produce red, you don't produce white. You produce a new character, which is pink. So neither of this is completely dominant over the other. So they produce an intermediate between red and, it means that this one has brought 50 and this one has brought 50, and then I form an intermediate color. Then if you look at the codominance, it means that the two must be there. If you look at this color and look at this color, the red and blue, when you cross them, you produce a mixture of red and blue. Here you produce a mixture. It means that red is completely dominant and blue is also completely dominant, and they express themselves in the phenotype of the offspring. While in incomplete, this an intermediate, because this one has brought me 50 and also this one has brought me 50. This one, you don't produce neither of the color from the parent. You produce an intermediate. You produce a new color. And then this one, codominance, together, it means that both of the parents' color is being expressed in the phenotype. I think that's how you can differentiate among the two, among the three. Then let's look at one of the questions involving the dominance. So they are saying that an allele for tallness is dominant over allele for shortness. A man who is homozygous, so I know that homozygous, tall, marries, and then they said, yeah, tallness is dominant over shortness. So it means that this guy is homozygous dominant. So marries a lady who is homozygous short. And then short, they say that is what is recessive, or it is recessive. And then using a genetic cross to determine the possible genotypes and phenotypes of the children. So what am I going to do? P1, parent 1, phenotype. Tall guy marries a short lady. You get it from the question, as I told you. From the question, a man who is homozygous, tall. This word homozygous will help us in the genotype, to identify the genotype. The word homozygous, heterozygous, only is important in showing which kind of genotype are you having. You don't put it in the phenotype, because you can't see that this person is heterozygous or is homozygous using the phenotype. You can't do that. You only know that this person is tall, this person is short. But if you go into genetic composition, you will know that, oh, you are homozygous tall or you are homozygous short. That's why we don't add the word homozygous or heterozygous here. So we are saying that a man who is homozygous, we have seen tall, yes, marries a lady who is homozygous short. So we go to genotype. Genotype. They said homozygous tall. Letters are supposed to be the same. And then you have homozygous short. Letters are supposed to be the same. Why they said it here? Homozygous short. Then you go to meiosis must take place. When meiosis takes place, it divides into two. Even here, it divides into two. What do you form here? You form what's called a gametes. So it means that this T is going to come here, this T is going to come here, this T is going to come here, this T is going to come here. And then fertilization must take place. It means that they must cross. You can use a cross or you can use a Punnett square. Either of the two is correct. And then they cross that, crosses this, and then that crosses that. So this one crosses with these two. That one is done. Then I go to this. This crosses with that, and then this crosses with that. And then I get the results, which you call the F1, the first period generation. So when I cross, I get this. So it shows that all of them were tall. But which kind of tallness? The tallness, if you are describing in terms of genes, it is heterozygous tall. So, or you can use this kind of option. Don't use 2 because we want 1. So then the genotype, it means that all of them are capital T, small t. Which kind of phenotype is this? Because I have the capital letter T, then it means that this individual is going to be tall. So which kind of phenotype? All of them are tall. I don't say heterozygous tall. I don't see the heterozygosity in the phenotype. I don't see that. So I just have to say they are tall. And then the individual of F1 all displays the dominant characteristic. It means that all of them are what? Are tall. In incomplete, you saw that in incomplete, you have the capital letter and the small letter. You see? You have capital letter and small letter. But in incomplete, neither of the allele is completely dominant over the other. Then it means that we don't have a small letter there. So an allele for red and allele for white is also dominant. When the homozygous red crosses with the homozygous white, it means that both of them are dominant. Using a genetic cross, determine the possible genotype and the phenotype of the flowers produced. I have P1 phenotype red. I get it from the question. Red crosses with the white. They are saying it. Red, white crosses with the red from the question. So I always get that mark from there. You have 3 marks free without even thinking. Then genotype. Now I have to come here and then look for these words. They're saying when a homozygous red, you see, homozygous red, same letters, homozygous white, same letters. Then meiosis takes place. When meiosis takes place, it also splits into gametes. So gametes, when they are formed, fertilization must take place. So you can use that method, or you can cross either of the two. So you bring these ones, you put them there. You bring these ones, you put them there. Then you cross them. You cross them. Or you can bring these ones here and then bring these W's on top. Either way, it's correct. And then F1 generation, if you look at them, all of them, they are capital R, small w. Capital R, small w. So the genotype, all of them is capital R, small w. And then the phenotype, which kind of phenotype is this? Capital R, small w. If you go back here, capital R and small w, it's not red, it's not white. So it shows that it is an intermediate of the what? Of the two. So it gives me pink. So if it gives me pink, it shows that this is incomplete dominance, whereby I produce a different color. And then offsprings have an intermediate form of trait of the parent. So that's how I can describe them. So note that in incomplete dominancy, you don't have small letters. Then codominance is the same as incomplete dominance, but the only change which is there is because you produce a mixture of the two colors. So they're saying the cow with white fur, capital W, were crossed with a bull with red fur, capital R, as shown in the diagram. Use the genetic cross to show how the run was got. So how did we get this? So phenotype, P1 phenotype, I have to look for it. White fur, so I say it's white fur crossed with red fur. So it means that red is crossing with white. Red is crossing the white. Here they didn't talk about homozygous, heterozygous. But the statement, the word "ron," shows that it's a mixture. Ron means a mixture of two colors. So it shows that these two were homozygous, so that they gave rise to this ron. So now I have to know that capital R and the capital R, small w, sorry, capital W and the capital W. Then when meiosis takes place, I produce gametes. Which gametes they undergo fertilization? I give you a tick and this. this and this. So it means that you have scored a tick there. And then I cross them. You can use the cross or you can use this method. Either way is correct. And then I get the F1. So P1 F1, I give you also a tick. So please don't forget those. Don't forget this. Don't forget this. Don't forget this. 3 marks are there for mahala, for free. So we are saying that the genotype is this. Why? All of them are like this. Capital R, small, capital R, capital W, capital R, capital W, all of them. So it means that this is not red, it's not white. What is it? It's a mixture of red and white, which we call ron. That's the meaning. And this is how they wanted you to do that question. It's not difficult if you look at it. In codominancy, you produce a mixture of the two colors. While in incomplete dominance, yes, you produce an intermediate color. But the crossing is exactly the same. So we are saying that both alleles are equally dominant and are expressed in the phenotype. Okay, this is just trying to do a summary. Proportion and ratio of the genotypes and the phenotypes. Sometimes they ask you to do the ratio, sometimes they ask you to do, yes, the ratio. So when you talk about the ratio, it describes the number of times a genotype would appear or the number of times the phenotype would appear. So for example, if you look at this, is capital R, capital R, capital R, capital R, capital R, small r, capital R, small r, and then small r, small r. So I just have to say this is 1, and then this is 2, and then this is 1. So I'm going to say 1 to 2 to 1. 1 is for this, and then for 2 for this, and then to 1. That's basically what they want in the ratio. And then if phenotypic ratio, you see how many. If this is let me see, they didn't give you the color. For example, if this is red and then small r is white, then it means that this one's gonna be red. This one's gonna also be red. Why? Because it is having a dominant allele which is masking this. So it's gonna be red 1, red 2, red 3, and only 1 white. So I'm gonna have 3, 2, 1. That's the meaning here. Actually, it's the same thing I've given you. Red is 3 red and 1 white. So the ratio is going to be 3 to 1. Note, let students practice different examples with the crosses. So it means that when I finish here, I'm going to send you some questions for you to practice. So basically, that's it. And then if you look at sex determination, we are saying that okay, let's look at variation. So differences among organisms of the same species, we call it variation. So when we talk about variation, we are talking about this organism. What are some of the causes of variation? These causes could be genetic or it could be environmental. You can be tall not because you are genetically tall. It could be environmental. You can be short not because you are genetically short. It could be environmental. Haven't you ever seen father, mother, both short, but they produce a tall kid? It could be genetical, yes, or it could be environmental. Oh, dark skin, or light skin, they produce a dark skin, and then the father said, this is not my kid. Who told you it's not your kid? It's whose kid? So it's your kid, but some environmental factors could happen, or some genetic factors could also happen. When we talk about a species, it's a group of organisms living together. And these organisms must interbreed to produce a fertile offspring. I've tried to explain this. A group of living organisms consists of similar individuals, meaning the same group, capable of interbreeding. Interbreeding is sex, having sex and produce kids. That's interbreeding. Right? Courtship, you could do the courtship and then you produce. I hope everyone is above 18. To produce fertile or fertile. Fertile means something which can reproduce again, or viable offsprings. Offsprings are babies. We saw them when you're talking about genetics. And then a population, if you have dogs, a lot of dogs in the same area, specific time, then we say that a population of dogs, population of plants. So when you talk about a population, is a group of organisms of the same species. They must be living together and they must be able to interbreed. So if they can interbreed, then it means that it's just a population of those organisms. The reason why I've talked about the species and population, because variation happens in the species, or it happens in the population. That's why I try to do a recap of population and species. What are some of the causes of variation? Number one, crossing over. You remember we talked about crossing over. When we talk about crossing over, homologous chromosomes come together during prophase 1. You must be specific, prophase 1, and then the chromatids of the homologous chromosome, they cross over. When they cross over, you'll find out that they produce chromosomes with mixed genetic composition. You find out that this part came from this chromosome, while this part came from this chromosome. So this one came from this, and then this one came from this. So it means that genes have been mixed. That's why you have the father, you have the same mother, but you have different people. They appear, they don't even look alike. Sometimes you go even somewhere and they say, like, hey man, you resemble someone I know there. Does it mean that your father went to that lady and then impregnated her and without knowing? No, it's because you share the same ancestor. Then it means that you still have those genes within us. So crossing over, it helps in when homologous chromosomes from form pairs during prophase 1. I said prophase 1 is very important of meiosis. Crossover can occur as you see it here. Crossover occurs, as I've tried to explain, leads to new combination of genetic material in new cells. So if they separate during anaphase, they will go to different cells. Then it means that these cells, they'll have different genetic material compared to the parents. If parents had this, then automatically the offspring, you will have this. So it means that you will have some characters from the father, some characters from the mother, but not all characters that you will be the same, exactly like the father. So crossing over helps in bringing about variation. Number 2, random arrangement of chromosomes. How do chromosomes randomly arrange? If you look at this, you'll find out that you can have this, maybe. Remember, they just do randomly. They are like chicken when they are going to mate. Okay, I'll talk about that. For example, we don't know whether this chromosome is going to be on this side or is going to be on this side. It can be like this color, and this color, or it could be like this. So we don't know which one is going to be this one, which one is going to be this side, this one is going to be this side. And remember, here we're just showing 4 of them. They are not 4 different positions, as long as they are homologous chromosomes, but they have different positions. So you end up having different combination of, for example, in this case, you're going to have this, you're going to have this. In this case, you're going to have this, you're going to have this, and then you have another combination, you have another combination. So you end up having many combinations. So random arrangement of chromosomes during metaphase result in the gametes with unique. You will find out that you have unique combinations of what? Of alleles. At the equator during metaphase allows different combinations of chromosomes or chromatids to go into each new cells, resulting in meiosis, making them different. So if you look at this sperm will be different from this sperm, and this sperm will be different from this sperm. All these, if you look at them, all of them, they are different, but it came from only 2 combinations, but you have many combinations. You have many combinations. So because of this, it will result, and you don't know which sperm is going to fertilize the ovum. So because of this, then it helps in bringing about variation. And then another one is random fertilization. Look at this egg, and look at these sperms. Look at these sperms. All these sperms, they are looking for this ovum. and we don't know which one is going to fertilize the ovum. You see, we don't know. So if it is like that, it is by chance, it's random. All these sperms are different, but we don't know which one is going to fertilize this. So if the guy produces like 100 million sperms, and then only one, and each sperm is different from the other, only one will fertilize the ovum. So what does it mean? It means that this time it will be a different sperm fertilizing. Another time it will be a different sperm fertilizing. Obviously, we will have different genetic makeup, and then we will have what you call variation. We will have some differences among us. That's why you have different colors, you have different heights, you have different sizes, you have different length of hair. You have why? Because of the genes are being mixed up during these processes. So you're saying that any sperm stand a chance of fertilizing an egg or cell. You see how fast your sperm was. I told you that you were lucky. How fast your sperm was to fertilize the ovum, so that you at least you can exist. If your sperm did not fertilize an ovum, and then another sperm fertilized, you think you would have existed. Someone else would have existed, not you. But your sperm was smart, was faster, everything was perfect, and then it was able to fertilize the ovum, and then you are existing. So you're saying between different ovum and then different sperm cells formed by meiosis, resulting offsprings that are different from each other. We saw that you have different sperms. Each sperm each cell is different from the other. So it shows that random fertilization can help in bringing about variation. Note, each individual is capable of producing over 8 million sperm. You see? Hey, do you know the meaning of 8 million sperm? If they give you 8 million rand, are you going to be rich or poor? Obviously, you're going to be rich. So now imagine that is sperm. Yeah, 8 million sperm are potential. When you talk about potential, it means that those which can fertilize, because we have other sperms which cannot fertilize. But random chance of any one sperm and egg coming together is a product of these two possibilities. Look, 70 trillion different combinations. That is the chance. You see how it means that you won a barrel of 70 trillion. 70 trillion. Even the budget of South Africa might not be 70 trillion. I don't know. I'm not a politician, but it might not be a 70 trillion. This is a lot of money. It's a lot of money if it is terms of money. But sperms, you have all these chances, but your sperm was able to fertilize. So it means that random fertilization can bring about variation. So it is also a cause of variation. Let's look at random mating. Random mating, you just mate whoever comes to your face. For example, if you are chasing this one and then someone comes, crosses your eyes, then you leave the other and then you go with this one. That is a random mating. Whatever comes to your face is what you mate. I think the people who make a random mating are the so-called, they call them mahoshas. They do random mating. Whoever comes, they go. What they're about is money. So that's what you call random mating. So mating takes place within species randomly. If you look at this, yes, this can mate with this can mate with this can mate with this. There is no restrictions that no, Mina, I want a tall, big, no. With this, no. Whatever comes to your eyes, you just go. That's what you call random mating. Between organisms within a species lead to different set of offsprings from each mating pair. So if I mate this with this, different set. This different set. Mate this with this, different set. So random mating also helps in bringing about variation. And then lastly, we have what we call mutation. Mutation means change in the genetic composition of an organism. This could be chromosomal. It could be a chromosome changing, or it could be the whole chromosome changing. Number of sets could change. For example, Down syndrome, you have an extra chromosome. so chromosomal mutation, or it could be a gene mutation whereby just a specific part of DNA just chops off or is inserted or is substituted, something like this. If you look at this, this is a substitution. This is a DNA. What happened? This was here. You see it is this color, the same color. But what happened? This cut and then another part joined here. So it's also a mutation. This will result in a different organism. These mutations could be beneficial or could be a lethal mutation. So changes the structure of gene or chromosome, and therefore the organism's genotype changes too. The moment you change the genotype of an organism, then automatically you change the composition, the phenotype of an organism. Remember, the genotype, the genetic composition, what is inside your DNA determines what is outside. How beautiful you are is determined by the genetic makeup. Not makeup. No. Not makeup. No. It is the genetics of your what is inside. Your manners, the way how you respond, the more how you think is being determined by this. However, it could also be environmental. It creates organisms with new, different characteristics from one generation to the next generation. So if there is gene mutation, if there is any mutation, if there is any change in the genetic composition of an organism, it will result in different organisms, which we call variation. Because the moment the organism phenotype has changed, then we say that variation has taken place. So mutation could be, number one, harmless. It can result in harmless. It has no problem with you. For example, you have an extra finger here. It's not changing your physical abilities. So it could be harmless. It could be harmful mutation. This one, it can even cause death. Albinism, it is a mutation, but it is harmful. How harmful? Because you cannot sustain yourself in sun. If you are original indigenous, like someone you know, yes, it means that. Then it means that you are strong. But if you don't have it, then automatically what happens? It means that you have a mutation, and then you won't be able to walk in the sun. And then you have the beneficial mutations. They can make you more advanced. Some of you watched these X-Men movies. Okay, that is a movie, but X-Men stuff, yes. They have beneficial, maybe this one can fly, this one can do this. Those are mutations, but beneficial to them. A mutation can be beneficial, or it could be harmful, or it could be just there without any problem with you. This variation could be a continuous variation, or it could be discontinuous variation. So let's look at continuous variation. Continuous variation, this is the type of variation where there is a range of characters. There is a range of characters. There is intermediate values. So if you look at this graph, you'll find out that you have a number of people, number of characters, and then the extreme ends, you have a number of them still, but the number of individuals which have the intermediate intermediate value, they are more than the two extremes. For example, if you look at height in humans or size, you'll find out that people who are too small, they are very few, and people who are too big are also very few. But people who are just in the medium size, there are a lot. So that's the meaning. That's why you see that at each extreme end, the graph will lower down, and then as you continue to the medium size or medium value, then you'll find out that the number is increasing. So if you skip or you pass the medium size or medium value, then you go back to the number dropping down until you reach 0. So basically that's continuous variation. We see that examples of this you can find out. You have the size, you have the height, you have the skin color. You have some people who are too dark, some people who are light, others that are intermediate. But you find out that those who are too dark and those who are too light, they are few. But those ones who are intermediate, you will find them that a little bit many. That's why you see them here, the same story. So this continuous variation can be controlled by environmental factors, or could be controlled by genetic factors. So, but the environmental factors has a big influence on that. However, even genetic factors are also part of it. For example, if you look at size among individuals, if you eat miserably, it means that you're gonna become fat. Therefore, it is environmental. It's not that you are born like that. It's because the way how you behave yourself, then it causes that you gain weight. So such characters which can be controlled by environment or influenced by the environment, then can be categorized as continuous variation. Okay, then you have what you call the discontinuous variation. When we talk about discontinuous variation, this one, the environment has a very small impact on it. It means that it's not being controlled by the environment. So in this case, you have characters that you find in the population has no intermediate values. For example, blood groups. You have blood group A, B, A, B, O, O, O. So you cannot have these blood groups. You cannot have blood group. You cannot be blood group A and you are blood group B. No. You cannot be in between there. No, you are either A, B, or O. You cannot be halfway or halfway A, halfway or halfway B. No. You either A, you are B, you are A, B, you are O. So it means that there is no intermediate in between there. But if you look at height, you'll find out someone is too tall, someone is next to there, another, the next height, like that. But for the case of blood group, it's not like that. Another example is tongue rolling. Yes, you can either roll the tongue if you try to see exactly. Some people, if you roll, you can roll the tongue. If you do like this, you'll find out that some people can do the tongue. They can roll the tongue, but others cannot roll the tongue. You either roll it or you don't roll it. So that is the meaning, that if you are discontinuous, then it means that you cannot, you are either this side or the other side. So these characters are being controlled by the genetic makeup of an individual. So if it is genetic makeup of an individual, then it means that these individuals, they either belong to one group or they belong to another group. The environmental factors cannot influence you. It cannot influence you to be blood. If you're blood group A, you cannot be blood group AB. If you are blood group O, you cannot be blood group AB because of the environmental factors. No, if you are born blood group O, you're gonna die blood group O. So if it is like that, then we say that the one which are being controlled by the genes, then we say that is discontinuous variation. So if you look at this graph, you will find out that you have A, there is no one here. There is no one here. You have B, there is no one here. You have AB, there is no one here. So it is discontinuous. It is not continuous like height. If you look at height, if you look at height here, you'll find out that for it is continuing.